A very good morning to everyone present here. On behalf of MIT Institute of Biotechnology, MIT University, Madhya Pradesh, Gwalior, it is our great honor and privilege to welcome all the distinguished dignitaries, eminent scientists, academicians, researchers, scholars, industry experts, and students gathered here today for the International Conference on Recent Advances in Biotechnology and Nanobiotechnology in BioNano 2026.
Biotechnology and nanobiotechnology are among the most dynamic and transformative fields of modern science, from precision medicine, vaccine development to sustainable agriculture and environmental remediation. These disciplines are shaping the future of humanity. In BioNano 2026 serves as a global platform where leading researchers and young scientists come together to exchange ideas, present innovations, and foster interdisciplinary collaborations.
Today's inaugural session is graced by a distinguished gathering of visionary leaders and eminent scientists whose contributions have significantly enriched the fields of science, education, and research. We now begin the formal proceedings by inviting our esteemed dignitaries to kindly grace the dais.
May I respectfully invite our Honorable Pro-Chancellor, MIT University, Madhya Pradesh, Lieutenant General V.K. Sharma, AVSM, retired, our Honorable Vice-Chancellor, MIT University, Madhya Pradesh, Professor Dr. R.S. Tomar, Guest of Honor, Dr. Shweta Gupta, Senior Scientist and Water Laboratory in charge, Delhi Pollution Control Committee, Professor Sadma Siddiqui, Professor in the University of Visa, Saudi Arabia, and Professor Dr. Vinay Devedi, Director, MIT Institute of Biotechnology, to kindly grace the dais.
Till yesterday, our chief guest, Professor Dr. Sangeeta Shukla, Honorable Vice Chancellor, Chaudhry Charan Singh University, Meerut, was gracing the ceremony, but due to an urgent meeting at Raj Bhavan, Uttar Pradesh, she is not able to attend the ceremony. We thank you, ma'am, for sending your blessings.
Every meaningful academic journey begins by seeking the blessings of Goddess Saraswati, the divine symbol of wisdom, learning, and enlightenment. May I now request our esteemed dignitaries on the dais to kindly proceed for the ceremonial lamp lighting, symbolizing the illumination of knowledge and the beginning of this scholarly gathering. Please, sir.
To express our heartfelt gratitude and respect to our esteemed dignitaries, we will now proceed with the ceremonial floral welcome along with presentation of a sapling symbolizing growth, sustainability, and nurturing of knowledge. May I now request Honorable Pro-Chancellor Sir and Honorable Vice-Chancellor Sir to kindly welcome our guest of honor, Dr. Shweta Gupta, Senior Scientist, Water Laboratory in Charge, Delhi Pollution Control Committee, Government of NCT of Delhi with sapling.
May I request Professor Dr. Vinay Dwivedi, Director AIB, to kindly welcome our invited speaker, Dr. Sadma Siddiqui, with us. May I request Professor Dr. Vikas Srivastava, Coordinator AIB, to kindly welcome our Honorable Pro-Chancellor, Lieutenant General V.K. Sharma, AVSM, retired with a sapling.
May I request Professor Dr. Raghvin Kumar Mishra to kindly welcome Honorable Vice-Chancellor Professor Dr. R.S. Tomar with a sapling. May I now request Professor Dr. Arvind Saxena to kindly welcome Professor Dr. Vinay Dwivedi, Director AIB.
It is my great privilege to introduce our Honorable Vice Chancellor, MIT University, Madhya Pradesh, Professor Dr. R.S. Tomar. Professor Tomar holds a degree of MSc, MPhil, and PhD and is a distinguished fellow of several prestigious scientific bodies, including FRSB, Royal Society of Biology London, FCVES Hong Kong, FICER, FSLSC, and FIASC. Professor Tomar is the Vice Chancellor of MIT University Gwalior and the former Vice Chancellor of IES University Bhopal. He was founding campus head of MIT Gwalior campus. He was the director and head of MIT Institute of Biotechnology, Dean Life Science, and Dean Academics, MIT University Madhya Pradesh Gwalior. Professor has more than 33 years of teaching, research, and administrative experience in higher education in varied discipline of science. Professor Tomar is actively engaged in teaching, research, and innovation. His research interests include nanobiotechnology, microbiology, herbal medicine, cancer biology, animal biotechnology, environmental biotechnology. He has to credit more than 155 research papers published in various national and international journals of repute. He has filed 28 patents, of which 24 has been published in the Patent Journal of India. 2 patents has been granted and 1 is under the process for grant. Professor Tomar has published more than 40 books published by the national and international publishers. Recently, a book entitled Nanobiotechnology, Concepts and Applications in Health, Agriculture and Environment, edited by him with co-editors, has been published by Apple Academic Press, CRC Press, Taylor and Francis Group, Florida, USA. He has delivered lecture in more than 90 national and international seminars, symposiums, conferences, workshops. He is the member of various national and international scientific bodies, reviewers, and editor of national and international journals. Recently, he has been elected as a fellow member of the Royal Society of Biology, London, UK. With his visionary leadership, MIT University, Madhya Pradesh continues to strengthen its commitment to academic excellence, innovation, and impactful research. May I now respectfully invite Professor Dr. R.S. Tomar, sir, to deliver the opening remark. Please, sir.
Thank you, Suruchi, Dr. Divya. Good morning to you all. Today we are organizing the 4th International Conference on Biotechnology and Nanobiotechnology, abbreviated as INT-BioNano. And for that, we have invited the people in their field having the expertise in their own domain. On the dais, you can see Dr. Shweta Shah. She is the CEO of BIPCOL. Dr. Sadma Siddiqui, the microbiologist, came from UAE. On the dais, Pro-Chancellor Dr. V.K. Sharma, the Director AIB, Dr. Vinay Dubedi, my friend, Professor Dr. Kaman Singh, who is sitting in the front row. Today, exactly, he's come for the one Viva OC of chemistry. He's the professor and head of chemistry department in Lucknow Central University. The directors of various institutes, HODs, HOIs, faculty members, and my dear students of biotech and pharmacy. You know, this particular conference, if you trace way back in 2011, when we conceived the idea of starting a department of biotechnology, and I was fortunate enough that it was me who started this department that time, and the first batch started with just 10 students. And today, when I see the strength of my students, I really feel proud of that, the initiative that took place 14 years back has flourished like a big tree. All these conferences, seminars, symposia, workshops that we organize, they have some themes and sub-themes. And the basic purpose is to invite the people all around the globe so that we can understand what exactly is going on in that particular field. And I think such kind of platforms are very important to understand each other. This is the fourth in series. We already organized 3 such kind of international conferences. And I remember the first one that we organized in 2016, in which we invited one professor from Rockefeller University, New York, Dr. Jargot. And you see that 2016 association with Jargot is still continuing. This is the basic purpose, that how we can exchange the ideas, not only ideas, we can exchange the faculty and students also. And we are doing that. So biotechnology, when biotechnology started during my graduation time, that time my professor were used to say, I don't know what is this biotechnology. Because he was the man of botany and he was given one module to teach in biotechnology. So he used to ask, tell me what is this biotechnology? Vectors, cosmids, plasmids, ये सब नए terms थे. And that time, it was not easy to understand this specialized branch of the biology, which is actually the amalgamation of different disciplines of the sciences that includes technology, engineering, mathematics, and of course, arts, humanities. Because a good biology student is that who is good in arts also, drawings. If you can't draw a good figure, then you cannot be a good biology student. You have seen your higher secondary time that some of the questions were put like this: draw a well-labeled diagram. That's all. And that well-labeled diagrams carries how many marks? 5 to 10 marks. We have seen that time. So that's why I'm saying that this particular field is the amalgamation not only of sciences, it includes the humanities and other aspects also. And with this, we can actually bring out revolution in the development of our big state or our country at large. Then came nanotechnology, nanobiotechnology, and this technology is going from fine to finest level. And we never know where it will end, because we have not yet explored the nanobiotechnology as such. Nano level, you all know, 10⁻⁹. And now it is gradually moving towards the further, finest details. that is pico technology, 10 raised to power minus 12. And scientists can foresee 10 raised to power minus 15 femtotechnology. What that shall be? Because today when we talk about nanobiotechnology, then we see the size of the hair. If you just compare it, then you can compare it as of this size. 100 to 10 nanometer, it will look like this. You can't see with the naked eye. Then how we will see femto materials? But yes, technology is growing, and that day will also come very soon when we shall be talking about femtotechnology. But my dear student, let me tell you, this is new for you, new for we all of us. But if we trace our roots, you will find that our researchers have already done the research of that level, where they were already talked about the nanotechnology. They already talked about the pico technology and also the femtotechnology. That is energy. Energy transmit from one place to another. You don't know what kind of energy is surrounding you. That energy is continuously emitting from you, going into the environment, and your own kind of replica may be there if they can be transformed into some physical form. This is what actually is hypothesized, and this is not hypothesis only. Our great gurus, the saints, the rishis, they have already realized this thing. Sitting in tapasya for long in the caves and moving from one place to another, a person to whom you are seeing in India, he is in New York also at the same time. How it is possible? So this is what actually the science has to do. It is very easily said that I don't believe in God because I have not seen the God. So what? You start learning mathematics with suppose only. When you prove any theorem, where do you start? Suppose this. So suppose God is there. Then you will realize, ultimately you will prove it. But God can't be proved in laboratories by simply finding out the God particle. God is beyond that, which only can be experienced with your own practice. Those who are practicing, they know the presence of God. Today only I was reading the Ramayana, and in Ramayana there was one chopai based on this, that when after 11,000 years of rule on this earth by Sri Ram, they were all leaving the Bharat. Not this Bharat, great Bharat, which we actually think of today. That Afghanistan, Pakistan, not only these, all other countries, they are there. Wherever you find some traces of Indian culture, they were all part of our great Bharat. And this was the great Bharat. And this is how we think that particular time when after 11,000 years of rule by Sri Rama, they are all leaving. Then he said, who is talking? That after 11,000 Ram leaving the Bharat, time everyone was coming to him and saying, sir, हम भी आपके साथ चलेंगे. Ultimately, सबसे लास्ट में हनुमान जी आए. हनुमान जी कुछ पूछा नहीं, और हाथ जोड़े खड़े हुए. तो हनुमान राम जी बोल के देख के बोलते हैं, तुम्हारी क्या इच्छा है, साथ चलना है या करना है? बोला प्रभु जैसे ही आप होने का आशीर्वाद देता हूँ. इस दुनिया में अभी बहुत कष्ट आएंगे. जामवन से बोला कि तुमसे त्रेता के बाद द्वापर में मिलेंगे जब मैं श्री कृष्ण अवतार में आऊंगा. हनुमान जी से कहा कि तुमको जब तक सूर्य चांद सितारे हैं तब तक इस दुनिया में रहो, क्योंकि लोगों को बहुत कष्ट आएंगे आगे चल के. तो त्रेता में लोग बहुत मजे में हैं. राम राज दुख काहू न बेअता. द्वापर में बढ़ेंगे और कलयुग में तो भरमार होगी. तो उन कष्टों को कौन हरेगा? तो तुम लोगों को साथ रहोगे और जो तुम्हें याद करेगा उसे कष्ट हरना। आज भी हम सब हनुमान चालीसा पढ़ते हैं और शक्ति का एहसास करते हैं। दिस इज द फीलिंग दैट यू फील टुडे। ये तो अपने सामने एक है ना उदाहरण, जैसे हम मैथमेटिक्स में पढ़ते हैं, सपोज दैट यू आर गेटिंग द स्ट्रेंथ एंड पावर एंड यू गेट इट। दिस इज प्रूवन। और हनुमान जी के लिए भी कहते हैं कि हनुमान जी खुद अपनी पावर भूल जाया करते थे, तो उनको भी आशीर्वाद दिया था कि जब कोई याद दिला देगा तो याद आ जाएगा। बहुत सारे ऐसे एग्जाम्पल्स हैं जिसमें आप लोग देखेंगे कि साइंस इज मिनिस्क्यूल, वेयर साइंस स्टॉप्स spirituality begins, and if we can prove it through the science, you can't prove. Science can be proven through spirituality, but not vice versa. So all these aspects, everything that is the part of science, that is a part of biotechnology, that is a part of nanotechnology, that is part of nanobiotechnology, all these fields are growing very fast, and they are not new to us because we are knowing them first time, but not that the knowledge was not there, it was there. So with this, I thank you all and wish you all the best, that we should have some good deliberations in this one-day international conference, and we shall be interacting with each other, developing some relations and contacts so that we can further advance in the field of nanobiotechnology. Thank you very much. Jai Hind, Jai Bharat.
Thank you so much, sir, for your kind words. Now, it's my privilege to introduce our Honorable Pro-Chancellor, Lieutenant General V.K. Sharma, AVSM retired. He is an illustrious alumnus of King George School, Shimla, and holds a BTech from the College of Military Engineering, Pune, an MTech from Pune University, and an MMS from Osmanian University, all completed with distinction. He was commissioned into the Indian Army Corps of Signals and served the nation with exceptional dedication of nearly 4 decades, eventually rising to the prestigious rank of Lieutenant General, the second highest rank in the Indian Army. For his distinguished service to the nation, he was honored with the Ati Vishit Seva Medal (AVSM) by the President of India. Apart from his remarkable military career, Lieutenant General Sharma has also been extraordinary academic administrator and visionary leader in higher education. He served as the founder vice chancellor of MIT University, Madhya Pradesh, where his leadership played a pivotal role in establishing a culture of academic excellence, research, innovation, and global collaboration. His guidance and strategic vision has contributed immensely to the growth of MIT University as one of the leading private universities in the region. May I now request Lieutenant General V.K. Sharma, sir, to kindly share words of wisdom with us.
Good morning to all of you. Good morning to all of you. Wake up. Just all of you, can you stand up once? Yeah, now sit down. Open your eyes and keep your mind open. Right. So Professor Dr. R.S. Tomar, the Vice Chancellor, Dr. Shweta Gupta, who is the CM of BIPCOL, Dr. Shadma Siddiqui, Dr. Kaman Singh, who is the HOD of Chemistry in Central University, Lucknow, all the HODs who are in front of me, and all the participants who are participating in this workshop, and my dear students. We have been organizing this workshop every year on nanobiotechnology, but year on year the perspectives keep changing. And this is one field without which I think the environmental cleansing which we are thinking of doing, the waste management, which is a problem today in the industry, the batteries which we are wanting to make last longer without charging, the electrical vehicles, the fuel which we want to be giving less pollution, agriculture where we want more production, organic materials to come, the relationship with regard to the environment of the nanobiotechnology, and of course, the food that we are eating, the modifications in the food that can happen because of the plants, and health, the most important factor in which nanotechnology plays a pivotal role by personalized medicine, by delivery of medicine at the right place, at the right cells, which are actually diseased, and in treating diseases like cancer or such like diseases which require specific medicine at specific points and not the brute application of force, which is being done through chemotherapy and other methods. So, to all these things, the single answer is biotechnology and nanobiotechnology combined. So, if we see today, there are the biggest problem that we face today, I think, is the shortage of energy, the health issues for everybody to be healthy, and the environment, the rate at which the global warming is happening, and the rate at which the pollution is happening, pollution to earth, that is the soil, which is leading to polluted growth of agriculture, pollution to water, which is leading to waterborne diseases, and our health getting affected, our lives becoming shorter because of that. The pollution to the air in the environment, because the air you breathe. Today's newspaper said that during the entire winter this year, not a single day, the AQI of Delhi was within the limits desired. Not a single day. People have been breathing poison in Delhi, reducing the life of everyone, especially the children and the elderly. They get affected because their immune system is weaker as compared to adults. But even adults get affected because of the in the lungs, and so many diseases happen which were not there earlier, which are now happening. So with such like things, the remedy lies only in biotechnology, nanobiotechnology. In China, they have been focusing more research than happening in India probably, and there they have invented a paint which is there on the buildings, which can absorb the carbon dioxide and some of the pollutants. That is one. Second thing is the fuels which are there have been so designed or are getting uh, designed in such a manner by adding some stimulants that the percentage of carbon dioxide and sulfur dioxide and sulfuric uh, gases which are in the exhaust, those are reducing. The electrical vehicles and the solar panels are reducing, trying to reduce the pollution in the air to quite a level. But the answer basically lies in having such structures which can automatically reduce the pollution which is happening. The worst is the water pollution. In Gwalior, if you would have read in the newspapers that the water which is being given by Nagar Nigam, and we are also getting water from Nagar Nigam only, that contains E. coli. Now, to what extent can the Nagar Nigam do about it? They are trying to treat it, but not to effect that it should be. During rainy season, the water stagnates, and on that, the mosquitoes flourish. As a result, diseases like dengue and malaria, they flourish. come rainy season and you find people lined up for malaria and for dengue in the hospitals. That got to be eradicated. There are countries where mosquitoes have been eradicated. There are countries where malaria has been eradicated. But we need to work on all this. My advice to all those who are working in this field is to do some practical research which can benefit the society at large and which can benefit our country and our nation and internationally also. As far as the government is concerned, the policies help in startups, the policies help in getting funds, and the Department of Biotechnology and allied agencies actively supports programs and nanobiotechnology to address national challenges in healthcare, agriculture, energy, environment, promoting translational research and innovation. And as far as agriculture, food security, and sustainable farming is concerned, it is all the more important. Nanobiotechnology is set to profoundly impact agriculture and food systems where resource efficiency and crop yields are paramount. Fertilizers is something, the fertilizers themselves, when given to the plants, we are eating those, in fact, because they get absorbed in the plants. And when you are eating poison, then our stomachs are getting upset. We got syndromes, bowel motion is not happening properly. We are not fit. And as a result, our livers are getting affected. Our kidneys are getting affected. People are having stones in the kidneys. All that happening because of the quality of material that we are consuming and eating. All this needs to be researched. And remember, when you do research, the research needs to be cost effective. It needs to be done in such a manner that it is implementable in a country and the research should be able to sustain itself and common man should be able to achieve benefits out of such research. So that is what it is all about. And my dear students, do not think that for research you are doing just BTech or you are doing BSc Biotechnology at your level. What research can be done? Wrong. Totally wrong. The ideas can come and you can still do research. There are lot many ideas, lot many papers are written, and the level of research which is happening in every field needs to be told by the instructors, by the teachers to the students that this is the level at which research has already taken place. If you think you can do something better than that, you should be able to do. The limits of knowledge must be known to you all in particular field in which you are now studying. You must know that this is already done and this is what we have to think about beyond that. Because if you don't think, you will not be able to innovate. And startups. There are many startups on this which are happening. Yesterday, you saw those of you who listened to the TEDx speakers, one of them mentioned that she's got a startup only on good, right? And that startup, because good is something which is indigenous to our country, which the farmers know about, and the benefits of this over sugar, which is white sugar, are very well known. Our ancestors, they used to take tea and in that they used to put good and not refined sugar. And I have also been from that time, I have tasted tea with good, which now has become a rare thing. And she done a startup on that, for which she got a funding of about 30 lakh rupees from the government. And from Shark Tank, she got a funding of 1 crore rupees. And she is the CEO of a company which she has launched from Chhattisgarh. So startups can be there on these things. And she, a young girl, a female, everything was against her. And she is the CEO of that company, which is having a good turnover. So think about all these things, innovate, do research, keep your minds open, and then sky is the limit for all of you. I am thankful to the organizers who have organized this workshop, which is uh, very much desired today to overcome some of the issues that I have just enumerated. So I wish this conference a great success and wish you all the very best. Stay attentive, listen to the speakers who are experts on this subject. So Jai Hind, Jai Bharat. All the best.
Thank you, sir, for your words of wisdom. Now, proceeding with our invited speaker, Professor Sadma Siddiqui. Dr. Siddiqui is currently serving as professor in the Department of Public Health at the College of Applied Medical Science, University of Bisa, Saudi Arabia. She holds a PhD in Applied Microbiology along with multiple academic qualifications, including MPhil and MSc in Microbiology, reflecting her strong interdisciplinary academic background. Dr. Siddiqui has an impressive academic record with more than 60 research publications, 9 book authored and several book chapters and intellectual property contribution. She has actively guided and supervised numerous postgraduate dissertation and research students, contributing significantly to academic mentoring and research training. Her research expertise spans microbiology, public health, infectious disease, and biomedical sciences. She has presented her work in more than 60 national and international conferences and workshops. May I request Professor Siddiqui to kindly speak few words with us. Please, ma'am.
Heartiest good morning, all of you. Respected dignitaries on the dais and off the dais, dear students, researchers, eminent scientists, industry person, and young scholars. First of all, I wish to thank you, Professor, Pro-Chancellor, Dr. Sharma, Vice Chancellor, Dr. Tomar, and Vinay Devedi sir, to inviting me and addressing this gathering today. Well, science has always been lightened that guide humanity through uncertainty towards progress. Today, as we are gathering for the international conference on the recent advance in biotechnology and nanotechnology, we stand at the founder discovery that has a potential to transform healthcare, the environment, and the future of itself. As Pro-Chancellor sir has said, you are the young generation. You have a power to change the world. So it is a great honor for me and a privilege for me to address the gathering at the opening ceremony of International Conference. I sincerely congratulate the organizer, the management of the university, the special thanks to Professor Dr. Vinay Trivedi sir and the Vice Chancellor sir for giving me the opportunity to address the students over here. Not last, but the organizing committee and the advisory committee has a thankful grade for bringing together leading scientists, academicians, researchers, and young innovators from the different part of the world to exchange their ideas, explore their latest development in the biotechnology and nanotechnology. Dear students, we are living in the era where science and technology are advancing at an unexpected pace. Biotechnology has already transformed medicines, agriculture, environmental management, and industrial process. At the same time, nanotechnology opened new frontiers for enabling us to work at the molecular level and the cellular level with an extraordinary perceptions. From targeted drug delivery systems and advanced diagnostic to the regenerative medicines and biosensors. These emerging technologies are refining the way we diagnose disease, design therapy, and improve the quality of human life. They are not only expanding scientific knowledge, but are also offering sustainable solutions to some of the most pressing global challenges. However, the true power of science lies not only in innovation. Do remember, the true power of science lies not only in innovation, but also in the responsibilities. Scientific advancement must always be guided ethical values and commitment to serve humanity. As Dr. Tomar said, our ancient knowledge, our ancient skill is hidden. You have a responsibility to explore that. बहुत इनरिच है हमारा एशियंट जो भी साइंटिफिक सिस्टम है, बहुत साइंटिफिकली डिफाइंड भी करा हुआ है. When you go through your back knowledge, you will find the way to solve today's problem. As a researcher and educator, our goal should be to ensure that the scientific knowledge contribute to the healthier, safer, and the more sustainable world. There are rare movements in the history when humanity stand at the crossroad, not of the uncertainty, but of the possibility. Today is one of the moment. From centuries, medicine has been reactive. We have waiting for the disease to appear before responding. But now, for the first time, we possess the tool to anticipate illness, to intervene earlier, and to personalize care at the level once unpredictable, unimaginable. Artificial intelligence is no longer confined to the computer lab only. It is decoding genome, identifying hidden disease pattern, and designing molecules. Biotechnology is no longer limited to the traditional therapies. It is editing gene, engineering cell, and creating biological solution to complex disorder. Nanomedicine is no longer theoretical. It is delivering drugs with atomic and enabling detection at the molecular level. Imagine when these three forces come together, something extraordinary happen. Hidden healing become intelligence. We move from the treating population to understanding individual, from standardizing prescriptions to prescribe target inventions, from hospital-centered care to continuous real-time health ecosystem. Imagine a world where cancer is detected before the first symptom, where cardiovascular risk is predicted year in the advance, where nano device monitor metabolic changes and alerts clinical physicians intensely, where AI design personalized therapies in the week rather than years. This is not a future of the present century. This is emerging reality of our generation. Yet technology alone does not define progress. Ethics, equality, and responsibility must guide innovation. Intelligent healing must be the inclusive healing. The benefit of AI-driven biotechnology and nanotechnology must extend beyond borders, beyond economy, beyond privilege. Conferences like this play a crucial role in strengthening scientific collaboration. They provide a platform where ideas are exchanged, interdisciplinary partnerships are formed, and young researchers are inspired to think creatively and push their boundaries of knowledge. My message is very simple to all participants, especially young scientists and the students. Let curiosity guide you. Let ethics shape you. Let innovation serve society. Science should not only advance knowledge, but also improving the quality of life for people around the world. The future of science depends not only on innovation, but also on the passion and the responsibility of the next generation of researchers. Transformation, we are the architect of it. The decision we make today in the research, in the collaboration, regulation, and the education will determine whether this coverage become a revolution for all humanity. Let us choose widely. Let us innovate responsibility. Let us heal intelligently. I am confident enough that this conference will lead to the insightful decisions, meaningful collaborations, and innovations idea that will contribute significantly to the advancements of biotechnology and nanobiotechnology, focusing on the global well-being. Once again, I congratulate the organizers and wish the conference great success. Thank you so much. I wish you all the productive and inspiring conference. Thank you, dears.
Now I would like to introduce our guest of honor, Dr. Shweta Gupta, Senior Scientist and Water Laboratory in charge at the Delhi Pollution Control Committee, Government of NCT of Delhi. Dr. Gupta is an accomplished scientist with over 2 decades of multidisciplinary experience in biotechnology, molecular biology, environmental monitoring, and vaccine quality control. She has played a pivotal role in strengthening environmental monitoring systems by implementing real-time water quality monitoring networks across several locations of the Yamuna River in Delhi, contributing significantly to environmental protection and pollution control initiatives. Earlier, she served as General Manager and Head of Quality Control at Bharat Immunologicals and Biologicals Corporation Limited, a Government of India enterprise involved in vaccine production, where she contributed extensively to vaccine quality assurance, regulatory compliance, and laboratory validation processes. Dr. Gupta also served as project head of the oral cholera vaccine project under the National Biopharma Mission funded by DBT BIRAC, further strengthening India's capability in vaccine development and evaluation. An exceptional mentor and academician contributor, she has guided and trained more than 500 postgraduate students, biotechnology trainees, and research scholars, significantly contributing to capacity building in biotechnology and pharmaceutical sciences. She is also associated with several scientific societies and has published research in areas including molecular genetics, biodiversity analysis, environmental monitoring, and vaccine research. May I request Dr. Shweta Gupta ma'am to kindly address the gathering.
First of all, I'm so glad and honored to be here. Thank you, sir, for having me here. I'm thankful to Dr. Tomar, Vice Chancellor, Dr. Sharma, Pro Vice Chancellor, Ma'am Siddiqui, and Dr. Vinay. See, I have been engaged as a student, as a scientist, as a general manager, as a head in science since 92. I did my graduation, did my post-graduation, then I have joined molecular biology, DNA fingerprinting unit, then vaccine field, then I have run a project that oral cholera vaccine, and ultimately I'm looking after Yamuna cleaning as a water lab in charge in DPCC. So I am the combo. You never know. Your student life will design you where you are going to be. But I have done my graduation from Gwalior. I have done my post graduation from Gwalior. तो आज मैं एक साइंटिस्ट की तरह नहीं बोलकर मैं अपने आप को आप में बैठा हुआ देख रही हूँ. मैं देख रही हूँ जब मैं यहाँ से चली थी तो कितने सपने थे मेरी आंखों में. बहुत सारे पूरे हुए, बहुत सारे एक्सपीरियंसेस लाइफ के मिले. मेरे अंदर एक स्ट्रॉन्ग टेक्निकल साइंटिफिक पर्सन भी है और मेरे अंदर एक अल्टीमेटली एट द एंड ऑफ लाइफ एक ह्यूमन बीइंग भी है. मैंने अपने सारे टेक्निकल एक्सपीरियंसेस से अपनी लाइफ के एक्सपीरियंसेस को जोड़कर कुछ जो भी निचोड़ निकाला है वो मैं उन बच्चों में बैठे हुए अपने आप को देखते हुए आज आप लोगों को बोलना चाहती हूँ. सबसे पहले तो सर ने बोला कि वी बिलोंग टू not India, from the great India. We all of us. सर ने बोला कि जागरूक रहो, सतर्क रहो. मैं भी यही कहूँगी, always be a student, be attentive. लाइफ जब सिखाती है वो आपको गूगल सर्च नहीं देती, वो आपको कोई बुक नहीं देती, रेफरेंस स्टैंडर्ड नहीं देती, कोई टेक्नोलॉजी नहीं देती, बहुत निष्ठुरता से सिखाती है। तो जब हम स्टूडेंट होते हैं, हमें तभी सीखना चाहिए, हमें अटेंटिव रहना चाहिए, हमें अपना एक गोल बनाना चाहिए, हमारे हाथ में प्रयास करना है। सो ऑलवेज बी अ लर्नर, इररिस्पेक्टिव टू whom यू आर लर्निंग। आप किसी से भी सीख सकते हैं, घर की मेड से सीख सकते हैं, रास्ते में जाते हुए जाम में सीख सकते हैं, आप बुक से सीख सकते हैं, लैब से सीख सकते हैं, और अपने मेंटोर से सीख सकते हैं। यू पीपल आर ब्लेस्ड कि आपको एजुकेशन मिल रहा है, और वो भी इतने प्रेस्टिजियस, इतने एक्सपीरियंस्ड लोगों के बीच में मिल रहा है। सो मेक यूज योर सेल्फ। व्हेन आई ज्वाइंड रिसर्च एसोसिएट इन 97, मुझे डीएनए फिंगरप्रिंटिंग यूनिट में चांस मिला। मुझे पीसीआर लगाने को मिलेगा, डीएनए आइसोलेट करने को मिलेगा। मैं सारी रात सो नहीं पाई, बस वो ही सपने थे, कैसे साइंटिस्ट, हाउ दे थिंक, हाउ दे वर्क, क्या बात करते होंगे, इस सब उसमें मैं सारी रात सो नहीं पाई। और जब मैंने देखा वहां पर, मैंने आइसोलेट किया डीएनए, आई kept so many PCRs with so many objectives। जब डीएनए मिला, दिखा नहीं हमें, बस पता था कि हमने ये प्रोसीजर कर लिया, ये हो गया। देन स्लोली स्लोली जब हमने उसकी क्वांटिटी बढ़ाई, या हमने टेक्नोलॉजी उसमें एड ऑन की, आई हैव सीन मोर देन 1000 डीएनए थ्रेड्स with my naked eyes in ultraviolet light and with the visual range। तो लाइफ में अगर आपका कुछ गोल है, इट्स लाइक डीएनए जो दिखता नहीं है, तो या तो अपने आप को इम्प्रूव करें, या टेक्नोलॉजी को समझें, या मेहनत की क्वांटिटी को बढ़ा दें। अल्टीमेटली आपके सामने रिजल्ट आएगा। जितने भी लोग यहाँ बैठे हैं, सब बच्चे सारे, मैं भी ऐसे ही बैठती थी, बड़े बड़े लोग आते थे, बड़ी बड़ी बातें करते थे, हम कंफ्यूज रहते थे, हम क्या कर पाएंगे, कुछ सीख पाएंगे, नहीं सीख पाएंगे। पर नहीं, मैं आज बच्चों की तरफ से एक बात बोल रही हूँ। सारे लोग बोलते हैं आजकल के बच्चे, वो ये नहीं है, वो संवेदनशील नहीं है, वो फलाना नहीं है। सर, हमारे हिंदुस्तान में कहते हैं, क्षितिजल पावक गगन समीरा, पंच तत्व यह रचित शरीरा। Being an biochemist, I can understand. I have unfolded DNA to the protein. Means you're controlling both to the expressive one। जब आपका पानी पॉल्यूटेड है, सॉरी आई एम वाटर लैब इंचार्ज, जब आपका पानी पॉल्यूटेड है, आपकी एयर पॉल्यूटेड है, आपका मिलावट खाने में इतना हो रहा है, हाउ यू कैन एक्सपेक्ट कि आप वही त्रेता युग वाले ह्यूमन बीइंग जनरेट करेंगे। अल्टीमेटली यू आर मेड फ्रॉम क्षितिजल पावक गगन समीरा, वो भी तो उतने प्योर होने चाहिए ना, तब तो वो प्योर प्रोडक्ट आएगा। तो आपका जो बायोटेक्नोलॉजी और जो नैनो बायोटेक्नोलॉजी है, दिस इज द स्ट्रॉन्ग टूल, एंड यू पीपल आर लकी इनफ, यू कैन हैंडल दैट, यू कैन अंडरस्टैंड दैट, you can revolve the world, you can change the thought process। it is only the thought, once you conceive the thought, आपके वर्ड्स, आपके एक्शंस, आपके बिलीफ के ऊपर चलते हैं। न्यूटन ने सही कहा था, ये सारा आइंस्टाइन ने भी सही कहा था, और हमारा सनातन भी यही कहता है कि अल्टीमेटली ये एक ऊर्जा है। आप उस ऊर्जा को अपने अंदर से पॉजिटिवली ड्राइव इन करिए। सर ने बोला, बिल्कुल सही बोला, स्काई इज द लिमिट। और आप तो नैनो तक जब जा सकते हैं, आप डीएनए देख सकते हैं, अनफोल्ड कर सकते हैं, यू कैन कंट्रोल। कितना प्राउड फील होता है एक मॉनिटर के सामने बैठ के आप सर्जरी को कंट्रोल कर रहे हैं, डायरेक्टिव्स दे रहे हैं। किसकी वजह से? आपकी जो भी ये टेक्निकल डेवलपमेंट हुए हैं उनकी वजह से। तो उन्हीं सबको यूज करिए। यहाँ कोई छोटे से नर्सरी क्लास में न्यूटन नहीं होता, आइंस्टाइन नहीं होता, आप आर्यभट्टा नहीं होता। हम सब में ही उनमें से कोई छुपा हुआ है। मुझे नहीं पता यहाँ बैठे हुए बच्चों में से कल कौन सा बच्चा क्या मैजिक कर जाए, उसकी लाइफ उसे कहाँ ले जाए। मैं आप सबसे कह रही हूँ, प्लीज बी अटेंटिव, ऑलवेज बी अ गुड लर्नर। अपने अंदर के टेक्निकल कैपेबिलिटीज को जगाइए, सीखिए, समझिए, जानिए। एंड एट द एंड ऑफ माई स्पीच आई वुड लाइक टू क्लोज माई स्टेटमेंट विद दिस: अल्टीमेटली आप कुछ भी बनिए, बट एक गुड ह्यूमन बीइंग जरूर बनिए। अगर आप कुछ भी अचीव करते हैं एंड यू आर नॉट अ गुड ह्यूमन बीइंग, तो आप कुछ भी अचीव नहीं कर सकते। यू ऑलवेज बी देयर फॉर सम ऑफ द अदर पर्सनस प्रॉब्लम। आप अपना अच्छा करिए, आप टेक्निकली अच्छा करिए। बायोटेक्नोलॉजी कहाँ से शुरू हुआ था, सर बोल रहे थे, कोई समझता नहीं था। मैं भी उसी दौर की हूँ। ओनली 5 सीट्स वर देयर फॉर बायोकेमिस्ट्री। तो और आज देखिए आप कहाँ हैं। आप यूनिवर्सिटीज भी उस पर डिजाइन है। आप भी, हम लोगों ने कच्ची सड़क को बनाया, आप लोग उसको थोड़ा सा हाईवे में कन्वर्ट करिए। आप उस पर अपने अपने थॉट प्रोसेस के हेलीकॉप्टर्स लैंड करिए, उड़िए एंड टच द स्काई। गुड लक। मुझे नहीं लगता आप में से बहुत सारे बच्चे मिलेंगे कभी कुछ अच्छा हासिल करते हुए। लुकिंग फॉरवर्ड, गुड लक, बेस्ट ऑफ लक। थैंक यू वेरी मच।
Thank you so much, ma'am. We now move towards a special moment of today's inaugural ceremony, the release of the conference souvenir of IntBioNano 2026. May I request our esteemed dignitaries on the dais to come forward. Also, may I request all the conveners and organizing secretaries of the conference to release the conference souvenir.
We will now proceed with the felicitation of our distinguished guests as a token of our respect and gratitude for their gracious presence and invaluable contribution. Now, may I request Honorable Pro-Chancellor Sir and Honorable Vice-Chancellor Sir to kindly felicitate our guest of honor, Dr. Shweta Gupta, Senior Scientist and Water Laboratory in charge, Delhi Pollution Control Committee, with a memento.
May I request Professor Dr. Vinay Devedi, Director AIB, to felicitate our invited speaker, Dr. Shadma Siddiqui, Professor University, Bisa, Saudi Arabia. May I request Professor Dr. Vikas Srivastav, Coordinator AIB, to kindly felicitate our Honorable Pro-Chancellor, Lieutenant GK General VK Sharma, with a memento.
May I request Professor Dr. Raghavin Kumar Mishra to kindly felicitate Honorable Vice Chancellor Professor Dr. R.S. Tomar, sir, with a memento. May I request Professor Dr. Raghavin Saxena to kindly felicitate Professor Dr. Vinay Dwivedi, Director AIB, with a memento.
As we come to the conclusion of the inaugural session, I would now like to invite Professor Dr. Vinay Devedi, Director, MIT Institute of Biotechnology, to kindly deliver the vote of thanks.
Respected Chief Guest, Guest of Honor, Honorable Pro-Chancellor Sir, Honorable Vice-Chancellor Sir, distinguished speakers, esteemed delegates, faculty members, research scholars, students, and all the participants. On behalf of Organizing Committee of the 4th International Conference on Recent Advances in Biotechnology and Nanobiotechnology in BioNano 2026, it is my privilege and honor to propose the vote of thanks. First and foremost, I express our heartfelt gratitude to our honorable chief guest, Professor Dr. Sangeeta Shukla in her absentia. I would also like to extend our sincere thanks to our respected guest of honor, Dr. Shweta Gupta, General Manager, BIPCOL, for her encouraging address and for highlighting the importance of biotechnology and industry collaboration in advancing scientific innovation. Our deepest appreciation goes to Lieutenant General V.K. Sharma, Honorable Pro-Chancellor, MIT University, Gwalior, for his motivating words of wisdom and continuous encouragement towards academic excellence and research innovation. I would also like to express my sincere gratitude to Professor Dr. R.S. Tomar, Honorable Vice Chancellor, MIT University, Gwalior, for his visionary leadership, inspiring opening remarks, and unwavering support in organizing this prestigious international conference. I would like to express my gratitude to our invited speaker, Dr. Shadma Siddiqui, for her address. I extend my sincere thanks to all the dignitaries on the dais for their presence during the lamp lighting ceremony, the release of conference souvenir, and the felicitation of our distinguished guest. My special appreciation goes to the organizing committee, the faculty members, research scholars, students, and technical staff whose tireless efforts, dedication, and teamwork have made this event possible. Finally, I thank all participants for joining us today and contributing to the success of BioNano 2026. Thank you very much.
With this, we conclude the inaugural session of BioNano 2026. We request all dignitaries and participants to kindly proceed for high tea. After the refreshment break, we will reconvey for the technical session and parallel presentation as scheduled in the conference program. Students of AIP will move to their respective department. Students of AIB will be gathered here after the tea break. Thank you. Ma'am, we will start within 5 minutes. Can you please give us that much time?
Yeah, sure, no problem. 5 minutes from now. Okay, I'll be ready.
Yeah, thank you so much, ma'am.
Okay.
पहले तो ये 4 क्लास नीचे रख दो.
Welcome back, all of you. We now move forward to the first technical session of the conference, Invited Talk Session 1, where we will hear from the eminent scientists who are making remarkable contribution in the field of biotechnology, healthcare innovation, and sustainable agriculture.
This session will be chaired by Professor Dr. Vinay Dwivedi, Director, MIT Institute of Biotechnology, MIT University, Madhya Pradesh. Professor Dwivedi, Director, MIT University Biotechnology, has done his schooling from Kendriya Vidyalaya No. 1, Kanpur. He did his BSc and MSc from Bundelkhand University, Jhansi. After MSc, he qualified GATE Life Sciences with All India rank 52 and completed his MTech in Biotechnology from Lucknow University. He was university topper in BSc, MSc, and MTech. He has completed his PhD in biotechnology engineering from Uttar Pradesh Technical University, Lucknow, and performed his research work in ICPO and ICMR Institute. He did his research in cancer biology. He has 23 years of teaching experience and research experience. He has published more than 60 research papers in various national and international journals. He has attended more than 30 international conferences and presented paper there. He has 12 patents and 5 awards in his credit, have also been a key speaker at over 20 national and international conferences. 4 scholars have completed PhD in his guidance and 4 scholars are continuing their PhD. His research of interest in nanotechnology, biotechnology, intellectual property rights, plant biology, entrepreneurship, and bioremediation. He is currently working as director, MIT Institute of Biotechnology, MIT University, Gwalior, Madhya Pradesh. Thank you, sir. So we request you to kindly chair the session.
It is now a great privilege to introduce our first invited speaker, Professor Dr. Jennifer N. Harikrishnan. Professor Harikrishnan is a distinguished professor in microbiology and molecular genetics at the University of Malaya, Malaysia, and has previously served as the director of the Center for Research in Biotechnology for Agriculture. Originally from the UK, she completed her BSc in microbiology from the University of Surrey and later obtained her PhD in molecular genetics from Carnfield University, where she was awarded the Chancellor's Gold Medal for the most outstanding graduate student. Following her doctoral work, she pursued prestigious postdoctoral fellowship at the University of California, San Francisco, and subsequently contributed extensively to the biotechnology research in Malaysia. Her research focused on molecular genetics of tropical crops, agriculture biotechnology, and the use of beneficial microbes to enhance crop resilience and productivity under climate change conditions. Her work has contributed significantly to the development of biotechnological strategies for sustainable agriculture and global food security. She has published numerous research articles in internationally reputed journals and has been actively involved in collaborative research projects addressing climate resilient agriculture and intelligent farming technologies. Today, she will be delivering her talk. May I now invite Professor Jennifer N. Harikrishna to kindly deliver her presentation. Please, ma'am.
Thank you very much for the kind introduction. Can you hear me clearly?
Yes, ma'am.
Okay, let me share my slides. Can you see the slides?
Yes, ma'am.
Yes.
Okay, let me put that into slideshow. One moment. Okay, are they in slideshow view now?
Yes, ma'am.
Thank you very much. And thank you very much for the introduction. Also, thank you very much to Professor Vinay Dwivedi for inviting me to join you this afternoon. And a very big thank you to all of you in the audience. I think there are a lot of students of biotechnology present. So I hope that you will find this talk interesting and maybe have some questions for me. So I'm going to cover quite a few different technologies, but they all relate to farming for food security. So I'll start with some of the key issues and challenges, why we need to farm intelligently, some of the things that we are doing at University of Malaya and some other technologies that are emerging around the world, and what we can expect in the future in smart farming. Just before I start, I will introduce the university where I'm speaking from. I'm in Malaysia, and people often ask, why is it University of Malaya if the country is Malaysia? And that is because the country is quite a young country, and the university is older than the country. The University of Malaya was established in 1905. And if you look at the map on the top right-hand side, Malaya is the region in the blue circle. But Malaysia actually was formed later and includes some states on the north of Borneo Island. So Malaysia is actually bigger than Malaya, but this university is based in Malaya and we keep the old name. And we are a very highly ranked university. We are the #1 in Malaysia and one high ranking in the world as well. So why do I want to talk about food security? We had many important areas for biotechnologists to be applying themselves in the future. But I choose food security because I think when we think of our life every day, we need food. So it's really one of the most important things we must have. And it is facing many challenges. First of all, let's look at global countries importing food. So Malaysia, as an example, is not very good at producing enough of its own food. And not only that, it's the cost of the food. Malaysian households spend almost 70% of their budget on food. This means it's very sensitive to rises in fuel prices like we're seeing at the moment with the conflict in the Middle East. We see that fuel prices are rising, food prices and fertilizer prices are rising too. And when you're already spending 70% of your budget on food, there isn't a lot of space for rising prices. Secondly, we have more people. The planet is getting more populated, and more people means we need more food. But we don't want to use more of the land because we need to also look after our biodiversity. We don't want to use too many more agrochemicals because we don't want to pollute the environment. And we don't want to use more energy because that is leading to global warming. So how are we going to produce this food? And we need to look at biotechnology for answers. We have also a growing population in cities. This is also leading to more problems of distribution of food. When people are living in the city, the food has to come into the city. So can we look at ways that we can use intelligent farming to produce more food in the city? Cities also are affected very much by heat. They become hotter faster than other parts of any country. And in fact, it has been advised that even a 1.5 degrees centigrade rise in global temperatures means we might need to shift what types of foods and crops we grow in order to feed everybody. And that means people will have to think about changing what they eat, and that's another challenge. Another one, yet still, is that farmers and people in general are getting older. So how do we manage farming if we only have older people? How do we attract young people? And how do we make farming possible for older people? Can we use technology to do some of the hard labor of agriculture? And while we are facing all of these challenges, whatever technology we use, we have to consider the sustainability and environmental stewardship. We still only have this one planet, and all the waste products, all the fertilizers and chemicals we use in agriculture end up polluting our planet. So we have to be very careful to produce the food in a way that is as clean as possible. So the definition of food security, and this is taken from the World Food Summit, was that food security meaning all people at all times have physical, social, and economic access to sufficient, safe, and nutritious food that meets their food preferences and dietary needs for an active and healthy life. So the two I've underlined, we need to make sure it's sustainable, all people at all times, must be able to have the food, and affordable economic access. I mentioned that for Malaysia, it's already limiting when you're spending 70% of household income on just food. So we should ensure that food is sustainable and affordable. And two very good reasons here. The World Bank is saying that actually agricultural development is one of the most powerful tools to end extreme poverty, boost shared prosperity, and feed the projected 10 billion people on the planet in 2050. And another good reason is we need to respond to climate change. There's an urgent need to transform agri-food systems and leverage their climate solutions. And this was from the FAO of United Nations in the State of Climate report. So when we're looking at affordability on the left-hand side here and sustainability on the right-hand side here, there are a few things we can do that address both. If we can decrease the use of water, fertilizers, and pesticides, that will decrease the cost and be more environmentally friendly. We also need to increase worker safety, and we need more reliable monitoring and management of the resources so we can use them efficiently. All of these both keep costs down and help an environmental sustainable solutions in agriculture. So this is where I come to my title. I said intelligently farming. And what I mean, we are all intelligent people. You're all in university. So that means you must be intelligent. How do we use this intelligence? How do we use our knowledge and our skills to solve these problems? One of the things we must do is consider how we farm. We must use the most appropriate technology. Now, the most appropriate technology is not always going to be the fanciest or the newest. It's the one that works, that is affordable, that is environmentally friendly. So we mustn't get carried away with new technologies, but we mustn't be frightened of using them either. And we need to use them in the most appropriate way. We have to consider what we are going to farm and where we are going to farm it. So this is a very long timeline just to show that technology isn't new. We've been using technology as humans to produce our food from the earliest prehistoric times. The early technology, you could say, was a fish trap or a bow and arrow. It's still technology, and it was the most efficient thing at the time when we were just hunting and gathering. As we began to collect seed and start early breeding, it allowed us to settle down into communities and populations to grow. And we enhanced the power of animals like horses and oxes to help. Machinery came along in the 1920s, and also antibiotics had been discovered and started to be applied in the food industry. The really big shift, the jump forward, was in the 1960s with the Green Revolution, when we had high-yielding crop varieties, irrigation, fertilizers, and more widespread mechanization. By the 1970s, we had single-ingredient herbicides. Remember these things. We're talking about the herbicides and the fertilizers. I'm going to come back to them. By the 1980s, we had our first genetically modified organisms as crops. And by the 1990s, the first recombinant vaccines, not the first vaccines, but the first recombinant vaccines that were used in farms. By 2000s, we had started our genome sequencing, and we had the first complete genome sequence of a plant, a model plant, a Arabidopsis, and mobile devices started to be used on farms. By 2010, we had the first examples of gene editing, and we now have some of those crops in the field. And 2020s, genomics and other sorts of DNA technology are widely applied in breeding and selection. Together with those, we now also see the introduction of machine learning, artificial intelligence, and cloud applied to farming as well. So they go hand in hand together, because genomics is big data. And then when we need big data, we also need the IT as well. So intelligent farming, using the most appropriate technology in the most appropriate way. So I know you're all interested.
you are not audible.
Well, you just have to unmute yourself. You are unmuted.
Okay. Can you hear me now?
Yes, ma'am.
Okay, let me try to go back to slideshow. How much did you miss? How many slides? This one was finished, right? Hello?
Yes, ma'am. After
Ah,
this.
after this. Okay. So I'm just going to skip forward to this one. So what I'm explaining on this slide is that when we talk about how we're going to use less land, one of the things we can do is do more of our farming indoors, controlled environmental agriculture, and for the outdoors, we need to use the land more efficiently. So this is where we can bring in technology for precision agriculture. So I mentioned, I'm not sure if you could hear when I said, although we are focusing on our biotechnology and our nanobiotechnology, we also need to use all these other technologies together to collect all the biological data and use it efficiently. So then we need sensors, we need AI systems for decision making, cloud computing and large data handling. We need to trace our food chain. This is for things like provenance and labeling, which enables us to know that the food is safe, and also for trading and export. And last but not least, of course, we have all the involvement of biotechnology, whether these are gene edited or genetically engineered crops, and things like biofertilizers and biopesticides. So one of the old technologies that is really emerging as a more scientific approach now, but I think if you talk to any farmer using traditional methods, they will tell you, we know this. The soil health is what is important. So the use of microbes in farming is not new. Last time it would have come from using animal waste as fertilizers full of good microbes for the soil. What happened was somehow along the way through the agricultural revolution, many people went to large-scale farming and they forgot about the principles of soil health. There were large areas of monoculture, there was intense use of synthetic agrochemicals, and this resulted in low biological diversity and poor soil health, which puts you into a bad cycle of then the soil is bad, so we need to put more fertilizers, so we're using more chemicals. It is expensive and it is not a good way to treat the environment. Now we have a much better understanding that yes, we do need some pesticides and fertilizers, but we also need to make sure we are not destroying the soil health, and we can actually understand the microbes better now. So what is new is we understand more about the microbes. We can study them, we can sequence them, we can see the diversity, and that allows us to balance chemical inputs together with the use of biofertilizers. There are already sets of microbes designed for particular stresses that will help for climate change, those that help plants survive in floods or droughts or salinity or heat stress. And we can also derive chemicals from the strains and use them as biopesticides and biocontrol agents. And right up to using just the RNA in RNA sprays, which I will talk about later, as well as using whole mixtures of microbes or small sets of selected microbes. One of the studies that we did in our university, where we got together with a few people from IT and physics and social science, was to come together to look at the effect of climate change, particularly hot heat in the cities, and how that would impact the growing of beans. In this case, our beans were long bean, which is a very popular vegetable in Malaysia and very easily grown in people's gardens. And we looked at the impact of the heat on the bruskula mycorrhizal fungi, AMF, during growth of the long bean. And to cut a long story short, what we found was that the hotter it gets, the worse the diversity of the microbes becomes. So we may need to help the microbes when we have hotter soils so that they can still protect the plants and help them to uptake nutrients and result in stronger, healthier plants. Another thing that we did, this was a different study, we thought that one of the places in Malaysia has various different ecosystems. And just like India, we have areas where we have a lot of mangrove on our coasts. And like India, there's a very high diversity of the biology in the mangroves, and especially among the microbes. So it's a challenging environment because the levels of salt and acid and the levels of water are always changing. So the plants that grow there rely a lot on having help from microbes that are able to survive in that environment. So we selected microbes that were growing on the roots of mangrove plants, and we selected strains with plant growth promoting activity. And then we put them on a plant that doesn't grow particularly in the mangrove, banana, and we challenged banana with seawater flooding. There was a specific reason for that, because we do have a lot of coast and it does get a lot of flooding with seawater, and that's going to get worse with climate change. What we found was that when we add these microbes to the banana plants, they are better able to withstand salt stress. And the same microbes actually also protected them against disease. In this case, we challenged them with Fusarium wilt, and it improved their resilience against these stresses. This would allow us to use less agrochemicals and to still maintain productivity against rising seed levels. Another combination of technologies is plant biotechnology, but together with electrical engineers. So we had a student from electrical engineering who was very interested in plants. He's actually the chairman of the Malaysian Plant Light Society. And we now can get light, LED lights in many different wavelengths. And so what he did was set up different lighting systems so he could have different wavelengths, different energy intensities, and also different time periods, and see what effects they had on the plants. So this first page here, we are looking at Hydrocotyle bonariensis, which is a type of pagaga. It's a similar herb used in a similar way to Centella asiatica, and but it is a different plant and it has a slightly different chemical profile. But it is a functional food. And what we found was when you change the light, you can actually improve the growth and you can get more of the desirable phytonutrients in the plant just by changing the light. It's already widely known that red and blue lights are important, but one of the things that we discovered through these studies was that other light, green light wavelengths and ultraviolet light also helped and in fact produced the best biomass and phytochemical yields when they were also included together with the red and blue light. And sorry, there's the name of the student, Narendran there. You can see he's the chairman of the National Committee of Illumination in Agriculture in Malaysia. One of the other things you can do, though, if you are using artificial light, is that you can use less energy. So in Malaysia, if we grow plants outdoors, we get a lot of pests and diseases. If we grow them in greenhouses, it gets very, very hot. If we grow them in artificial light with white light, they also get pretty hot. But if we use LED, where we're just using the wavelengths we need, it doesn't get so hot. So we don't need to use so much air conditioning. And we can use less energy overall. So in this experiment, he compared the use of greenhouse, white light, and then just the LED lights. And the plant we used for this one is Stevia robunia. You might know stevia as an artificial sweetener, a natural sweetener made in plants. And what we found was that we could optimize the energy and produce the largest amount of those particular sweetener compounds in the plants through this technology. I'm going to mention this. This was not our work, I'll make clear. This was work that won a Nobel Prize. We didn't win that. That was won by this group here mentioned, Mungi et al. And they use quantum dots. And these quantum dots actually can be applied to greenhouses and used to modify the light wavelength. So instead of using LEDs, you can also use greenhouses with quantum dots to change the light wavelengths and improve the efficiency for crops. In this case, they were using tomatoes. And this is an example of a nanotechnology applied in biotechnology. So I thought you would be interested in this. We also have a group in our sister university in Malaysia who are applying the quantum dots directly to the plants and also seem to get better light use efficiency with that method. To mention UV again, but this time UVC radiation. This is very high energy wavelength, and it is very damaging actually. So normally it doesn't affect us because it doesn't get through the atmosphere of the Earth, but we can produce it artificially. And it has been used as a technology to prevent post-harvest spoilage in plants like tomato. But in this study, what we did was we used the UVC in a very low dose just to trigger a stress. And this actually increased the expression of flavor compound genes in the fruit. And we could determine that not just by taste testing. Of course, we do taste testing because we want to know, do they really taste better? But also they were actually sweeter and had different phytochemical profiles. So you can use these kinds of extra technologies as a kind of sister to biotechnology to improve fruit quality. And this is, again, so far, I've managed to say we don't have to do any genetic modification. This is just using the environment. But what if we do want to start having a go at changing gene expression in a different way? So this is another technology, RNA, and RNAs can be increased by genetic modification technology. But another strategy is to not do a modification of the plant, but just to treat them with RNA in a similar way that the RNA vaccines work. So what happens here is you spray double-stranded RNA onto the plant, and it goes into the cytoplasm. And it actually then will target and silence the genes with complementary sequence. So actually working at the level of RNA interference. And this can be used in crop and animal health as well as trait improvement. So this is some work that also does involve nanobiotechnology because RNA itself can be quite unstable. So one of the things that people do is to use nanocarriers. And in this case, we are using peptides, and we mix them together with the double-stranded RNA to improve the efficiency of their use. So what have we done with this technology? So our first model was to actually use it against plant viruses. In this case, it's not food, it's orchid, although there are food orchids. Vanilla is actually an orchid. But in this case, we were just using the ornamental orchids. And when we spray with the double-stranded RNA targeting the virus, we can actually interfere with the virus life cycle and protect the plants against the virus. The same technology does work on food crops. This is another example where we're using it on melon. And in this case, we're also targeting a virus. So if you look at the red box, this is the leaves at 10, 20, and then 30 days after infection with the virus. Inside the red box, they're just infected with the virus and not treated with RNA. Hello?
Excuse me, ma'am. Can you please wind up a little early as it's next speaker is waiting for us.
Okay, but I started very late. I have not had half an hour.
Yeah, thank you so much.
So you want me to stop now?
Now you can continue. Let's just wind up a little early.
Okay, I maybe can skip a few, but basically this is just to say this technology can protect the plants. And this slide is not our work, but this is the first commercial RNA spray. It is now being used in fields to protect potatoes against beetles. So not just viruses, it can actually work against insects. And it's actually seems to be quite acceptable also to the farmers, even the beekeepers, because a lot of pesticides actually harm bees. But this one is completely safe because it only targets the beetles and doesn't affect the bees. And we can also actually use RNA to change other traits. In this case, the bottom row is what the normal flower development of the orchid flower is. And the 3 rows above are different recipes of RNA that have changed the color and also the shape of the flower. So we can do this in a non-genetically modified technology just by spraying RNA onto the plants. And the last one is just to mention that if you do gene editing, you can do very precise changes. The chickens is not our work, but it is actually one that has shown a very good protection against bird flu. And we're applying a similar technology in DNA-free gene editing in banana now. I hope you give me time just for this last one because I really like this study. This was done by one of my students, Jasdeep, who was actually attached for part of her study to the ICGB in New Delhi. So she spent some of her project time in India. And what she did was we take the banana leaf after the farmer has harvested the fruit, and we can bring the leaf into the lab and use it to produce proteins. In this case, it was an antibody against toxoplasmosa. So the plants are not genetically engineered. You still get your crop of banana, and then you can use the waste product of the leaf for producing a high-value protein in the laboratory. So this would be another way that farmers could improve their income, and it doesn't have an impact on the environment. This one is just showing how much biotech crops are spreading, and this one just to say that the general consensus from scientific societies is that this technology is completely safe. And the World Food Prize in 2024, it was said that genetic engineering and gene editing technology are really important. They do more than just improve crop yields. They bring people and cultures closer together. And this just to summarize where the technologies are going in the future. So you can see biotechnology and nanotechnology are going to be really important together with the use of engineering, electronics, and information technology. So we have challenges ahead to make sure we can sustain food security. We need to use intelligence to improve crops, and biotechnology is a very big part of that picture. And nanobiotechnology is going to be really important to making sure that we can have an innovation-led farming culture and feed everybody on the planet. This is some of our teams and partners for, I talked about very many studies here, so it involved a lot of different people. And thank you very much for listening.
Thank you so much, ma'am. So the house is open for questions. If anybody has any questions, they can ask. Ma'am, any questions? Yes, please.
Yes, I'm happy to take questions.
So good afternoon, ma'am. Thank you for such an interactive session. As you already mentioned that the UVC has some damaging effect, and we already have studied that the blue light can trigger the oxidative stress. So what is the harmful effect in farming of the blue light that you are already using?
Okay, thank you for the question. No, the blue light is not harmful. The UVC is harmful, but we use it for very limited exposure to the plants. For the blue light, it's good, but we found that if we use it in combination with the green and the UV light, we get better crop productivity.
Okay, sir. Okay, ma'am. Thank you.
Any more questions?
Thank you, ma'am, for the very informative session. I have a question. I am working on plant growth promoting rhizobacteria. So what type of technologies we should opt for improvement of tolerance in legume crops?
Thank you for the question. So you probably will have some nitrogen fixing ability in the rhizobacteria that you're looking at. Many of them do. And what I think the optimal way to use them is actually to combine a few different strains together that maybe have different strengths. So you will want some with nitrogen fixing ability. You will maybe want to mix in some that are good at solubilizing phosphate. And you may also want to screen them for their ability to be antagonistic to different pathogens. So if you're talking about your legumes, you need to look at what are the pathogens in your legume crops, and you can screen to find members of your rhizobacteria community that actually help to protect the crop. And we found that if you can, yeah.
Okay. Thank you so much.
Thank you for the question.
Any more questions? Okay, thank you, ma'am, for this insightful lecture. We thank you so much.
Okay.
So now move on to our next lecture. I would like to invite Professor Dr. Shadma Siddiqui, ma'am, for her talk.
आपको माइक काटना चाहिए।
Good morning, all of you. So, after a tea break, how you all feeling? Good? Okay. Basically, I am an academician. I'm educator. Today, just I want to flow an idea with you all. The young scholar should get this idea. Well, thank you so much for giving me the opportunity to discuss an idea for a young generation. We are living in the era where biotechnology is no longer confined to the laboratory. So myself, Dr. Shadma Siddiqui. Dr. Shadma Siddiqui, I'm presently affiliated with the University of Bisha, Department of Public Health, in the Kingdom of Saudi Arabia. In my university, we are working on artificial intelligence. We are collaborating with other sciences, satellite sciences, and we are working on the artificial intelligence in healthcare. So I thought that I should share with you all what is the benefit of artificial intelligence in healthcare premises. An extraordinary junction in human history where intelligence is no longer solely biological, and the medicine is no longer bounded by the traditional limitations. What we are witnessing today is not merely a progression of technology, it is a transformation of healing itself. We are living in the era where biotechnology is not confined to the laboratory, it is transforming healthcare, agriculture, environmental sustainability, and industrial productions. The coverage of biotechnology with nanoscience has accelerated discoveries beyond imagination, opening unprecedented avenue for precise medicines, sustainable development, and advanced diagnosis. Today we gather here to understand and today we gather here the moment of extraordinary scientific transformation where advances in biology are being propagated forwarded by the innovations in nanoscience and artificial intelligence. So today I will discuss with you all how intelligent healing can be done. The coverage of AI with the biotechnology and nanotechnology. Artificial intelligence has emerged a disparate force in the life science, enhancing how we discover drugs, decode genome, engineer organisms, monitor ecosystems, and design nanomaterial. AI is no longer just a tool. It is a partner in the scientific discoveries. If we thought history, revolutions in the medicines has followed technology breakthroughs. For example, invention of microscope opened the microbial world. Invention of biotechnology transformed infectious disease management. Molecular biology decoded life at the genetic level. Today we stand at the next generation where computers meets biology and nanoscale engineering meets medicine. Artificial intelligence is no longer an auxiliary tool. It is becoming a co-investor and a diagnostic assistant and an event of molecular designer. You can see over here, artificial intelligence, once an abstract concept, has now become a cornerstone of the biomedical innovation. In 2023, the global AI drive biomedicine economic exceeded more than $2.5 trillion, involving more than 7,000 companies and thousands of researchers worldwide, and its project to grow towards 6 trillion USD by 2007. This statement has given by the CNN. Simultaneously, the AI in the biotechnology market value are roughly USD 5.6 million in 2025. Is forecast to be nearly 27.43 billion by 2034, growing at the compound annual growth of 19.3%. This research has been stated in the proceedings. This explosive growth reflects the acceleration adoptions of computational tools across research and development pipelines, from genomic to drugs discovery. If we talk about the data, biotechnology and AI has together unlocked capacity that were science fictions a decade ago. Today, algorithm can process terabytes of genomic data in under 25 hours, enabling insight at a scale impossible for human analyze alone. This statement has been written in CEO sandwich. In oncology, for example, AI models has increased diagnosis accuracy in hereditary cancers by more than 20%. And near about two-thirds of the personalized treatment plan now level AI algorithm for optimizations. This is not incredibly improvement. It is a paradigmic shift. So, traditional discovery timelines of 4 to 5 years are now compressed to 12 to 18 months. AI can screen 200 million compound per day, comparing to 2,000 throughout conventional method. Clinical trials failures rate have been dropped by approximately 15% with AI assisted design. These are not just a figure representing efficiency gains. They are signal lives saved, suffering reduced, and hope accelerate. These figures are not economically, they are reflecting scientific acceleration. But beyond marketing projections lies a deeper transformation. Medicine is becoming predictive rather than reactive. Now, nanomedicine meet intelligence. While AI provides the brain, nanotechnology provides the hand, target, prescribe, and transformative. Nanomedicines enable smart drug delivery systems, biosensor capable of single molecule detections, and adapting nanoscale therapy that respond to biological signal in real time. Today, researchers in AI integrated micro and nanobots are already demonstrating autonomous navigation through the complex biological environment for diagnostic and therapy. This coverage is a higher of the future where treatment is not just precise, but intelligent and adaptive. So what is the impact of global health system? What is the impact of AI on the global health system? The integrating of AI, biotechnology, and nanomedicine is reshaping healthcare beyond laboratories. AI-enhanced diagnostic tools are enabling earlier detection of disease like cancer, where early diagnosis can increase survival rate dramatically. Predictive analytics are helping health system intercepting outbreak and optimizing resources allocations, an essential capability highlighting during the COVID-19 pandemic. We have witnessed this. Wearable, smart, and connective nano devices are supporting chronic disease management in real time, bridging the gap between the clinical inventions and everyday life. So let us discuss some of the applications of AI in different biomedical fields. Number one, in oncology. Earlier cancer detection, AI assists earlier cancer detections and radio interpretations. Nanoparticle-based targeted drugs delivery. Precision oncology using genomic and molecular profiles. AI models for predicting tumor progressions and treatment response. If we talk about the cardiovascular medicines, AI algorithm for early detection of heart disease from ECG and imaging, nanotechnology for targeting drug delivery in arthritis, biosensor for real-time monitoring of cardio biomarker, regenerative biotechnology for repairing damaged heart disease. Regenerative medicines and tissues engineering, stem cell engineering guided by the AI analysis, nanomaterial for tissue regenerations, AI drive optimizing of biomaterials and cell growth conditions, bioprinting of organs and tissue using nanomaterials. All these are guided by AI drives. If we talk about the infectious disease and immunotherapy, AI-based pathogen detections and outbreak predictions are going on. Nanoparticles, vaccines, and drug delivery systems are on the way. Biotechnology for rapid vaccine and therapeutic development. Immunoprofiling using AI for personalized immunotherapy. If we talk about in the neurology and degenerative disorders, AI for early detection of Alzheimer's and Parkinson disease, nanocarriers for drugs delivery across the blood-brain barriers, neuroprocessing integrated with AI with functional restoring, biomarkers discovered through genomic and proteomic biotechnology. And in the field of genomic and precision medicine, AI analyze for large genomic database, based gene editing technology, personalizing therapy based on the genetic profile, nanobiosensors for rapid molecular diagnosis. In the pharmacy and the drug discovery, AI accelerate drug discovery and molecular modeling. Nanobiomedicine based targeting drug delivery systems, biotechnology platforms for biology and the molecular antibodies, predictive AI models for drug toxicity and efficiency. So these are the biomedical field where AI playing a vital role, and in today's world, the healthcare system is changing. The coverage of AI and the biotechnology should reduce disparities, not widen them. As a scientist, we must campaign ethical AI framework and interdisciplinary collaborations. So whenever we talk about the innovation, there should be a collaboration. Interdisciplinary collaboration will always help to become the molecular, to success in any of the research theme. AI changed the healthcare system from reactive to predictive medicines by 4 Ps. Predictive, prevention, personalized, and participations. These are the 4 Ps by which AI playing a role in healthcare systems and changing the method of diagnosing, method of detections, method of treating the patient, and doing the personalized medicines. What can be the next vision in the upcoming 10 years? AI design personalized nano therapy, fully digitalized clinical trial ecosystem, real-time biosensor drive treatment adjustment, autonomous nanorobots drug delivering prototypes, AI integrate synthetic biological circuits also. Beyond the boundary, digital intelligence and biological system will continue to dissolve. Healing will become intelligent and adaptive and anticipatory. At last, I just want to conclude. AI, biotechnology, and nanomedicine is not just reshaping the healthcare, it is redefining our relationship with the health system. This intelligent ecosystem will lead to predictive prevention, personalized and participating medicines. Medicines that anticipate disease before it manifests, tailor therapy to be individual and empower patients with real-time insight into their own biology. This conference represents not just sharing of the knowledge, not merging a scientific event, but also collaboration committed to harnessment, biotechnology, and AI for the betterment of humanity and healthcare system. Thank you so much. Do you want to ask any question, dear students? I just want to throw an idea in your mind. It should be clicked that whatever you are planning for your research work, it should be collaborate with AI also, because future is based on AI. You should not you should not be in the hand of AI. You should master the AI. Okay? Be careful, and you should follow the ethical rules and the commitment towards the humanity. Thank you so much. Can I take a-
Thank you, ma'am, for your talk. Now, I will introduce our next speaker, Dr. Emalda Chukwu. She is a senior research scientist at the Center for Infectious Disease Research, Nigerian Institute of Medical Research, Lagos, Nigeria. She is a distinguished medical microbiologist with more than a decade of experience in infectious disease, epidemiology, and antimicrobial resistance research. Her research focused primarily on respiratory and diarrheal diseases in children, antimicrobial resistance, surveillance, and antibiotic stewardship using the One Health approach. Dr. Chukwu has been actively involved in several major international research initiatives, including projects focused on vaccines to control respiratory pathogens and antimicrobial resistance across Africa, as well as national program aimed at improving child health outcomes. Her outstanding scientific contribution has earned her several prestigious recognition, including the Norbert Elias Fellowship at the University of Bielefeld, Germany. She is also an affiliate fellow of the African Academy of Science and a member of the Global Young Academy and the Nigerian Young Academy, reflecting her leadership in the global health research and antimicrobial resistance surveillance. May I now invite Dr. Imelda E. Chukwu to kindly deliver her presentation. Please, madam.
Ma'am is joining in a while. Let's wait. Okay, so we are coming to the end of the session one of the invited talks. I would sincerely thank all the distinguished speakers for sharing their valuable insights and knowledge with us. And now we will take a break for lunch, and the next session will begin as per the conference schedule. We request all the participants to kindly resemble back here in the afternoon technical session. All students, you will all gather here again for the second session. Thank you so much. You can move for lunch. इधर 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 Thank you.
TED, we have background, global context, and biotechnology, rapid diagnostics, advanced diagnostics for AMR, precision diagnostics, nanotechnology-enabled therapeutics, then the challenges and translational barriers. So by way of background, we already know that infectious disease continues to be a major challenge to public health, particularly with the growing threat of antimicrobial resistance, which has exacerbated the issue of infectious disease. So AMR naturally occurs when bacteria or microbes evolve in such a manner that they are able to evade the drugs that are targeted to eliminate them. So that is the mechanism they use to survive antimicrobial drugs, thereby rendering conventional treatments less effective and contributing to millions of deaths worldwide. So this crisis is being compounded by emerging pathogens, both the emerging and re-emerging pathogens, and also biofilms that are associated with infections. This shelters bacteria from antibiotic penetration and immune clearance. So all these are further complicating treatment outcomes. So now, when as this resistance is becoming more pervasive, the drugs that we are using currently to treat them are no longer effective. We're fast running out of options for treatment, and the drug pipelines are thinning down very fast. So there is a risk that modern treatments that we're currently adopting, like chemotherapy, transplantation, may not be feasible again, because the risk will outweigh the benefits. That is the risk of infection with resistant pathogen and untreatable infection when it becomes higher than the benefits. So it may not be attractive anymore as a means of therapy. So what is the global context? Globally, we know that AMR is a problem, and it's been predicted that there will be about 10 million deaths annually per year by 2050 if nothing is being done to curtail or mitigate the current trajectory. So, and even the emerging pathogens are not helping matters. We have pathogens like SARS-CoV-2 that just emerged, and other re-emerging pathogens that are also worsening the system, making it even more difficult to tackle the existing public health threats. And again, the traditional antibiotics and the diagnostics we are using is lagging behind, because we are fast running out of options. The treatment options we have, drug discovery pipelines are slowing down, and while resistance is emerging faster than the discovery of drugs. So we have a situation at hand, and this generally impacts public health. And then by increased hospital stays, and then healthcare costs is increased, it also affects the food and agricultural sector, causing threatening food safety and food availability. So then what is the innovation now? The innovation here is that to address these critical challenges, biotechnology and nanobiotechnological innovations are driving revolutionary interventions that go beyond the traditional antibiotics that we're used to. So what happens is that it enables precise diagnostics, so you can have targeted treatments, advanced therapeutics, and innovative delivery platforms. So all these collectively strengthen our fight against resistant pathogens and emerging pathogens as well. So in the biotechnology area, we have that rapid diagnostics. And the key innovations in this area is the availability of next generation sequencing platform for surveillance, I'll talk more a bit about that as I go forward. Then we have the CRISPR-based detection, which has revolutionized our ability to do gene editing and modify resistant pathogens to become susceptible. Then we have more recently the artificial intelligence assisted pattern recognition. So all these have benefits, all these innovations have benefits because they allow rapid detection of disease pathogens, and it does that with high sensitivity and specificity, being able to target the exact pathogen that we are looking for. So now looking closely at the NGS. NGS is an advanced DNA sequencing technology that allows rapid and high throughput analysis of entire genome. So the beauty of it is that you can analyze the entire genome of a microbial community or a specific microorganism. And then you're able to use this opportunity to analyze. You can analyze several DNAs at the same time, unlike the traditional sequencing method. The next generation sequencing method allows you to analyze several fragments of the DNA at a time. And you can sequence millions of DNA fragments simultaneously. So the key steps that you take in that is first, the key applications in AMR and infectious disease surveillance is, yes, you can use that to detect use that to detect antimicrobial resistance because it gives you resistant genes, because it gives you a comprehensive approach. You can look at the whole genome of a bacteria or a virus, and then you can see all the genes that are available that are causing resistance. So you have all the resistant genes, and you can look at the ones that are available responsible for antimicrobial resistance to a particular antibiotics. And then you can also use that to check for virulence genes as well. So it can also be used for genomic surveillance of infectious disease in real time. You can use that to track infectious disease outbreak, analyzing the genetic similarities among these pathogens. Then another application, a major application that is now commonly used widely, is the application in wastewater surveillance, environmental surveillance. You can apply this next generation sequencing, especially the metagenomic sequencing. It allows you to look at the microbial communities available in wastewaters and in the environment, such that you can even determine the ones that are going to be outbreak prone or pandemic prone pathogens that are being incubated in the environment. So you can, in this manner, predict future outbreaks and then can now take a proactive measure to ensure that you mitigate that outbreak. So it's been widely used now for surveillance purposes. Then we have the molecular epidemiology, transmission mapping. So you can use this high resolution data that it produces to map related microbial strains and then look at evolution, disease evolution. So now, a well-known example where this has been used widely was during the COVID-19 pandemic, where sequencing data helped us to identify the emerging variants. Even as new variants were emerging, sequencing allowed people to keep tracking. We track the transmission strains, inform public health interventions. So these are some of the ways it can be used as an outbreak prediction tool. So similarly, it can also be used to monitor outbreak of pathogens. So it's not just COVID-19, SARS-CoV-2. But it can also be used or has been also used in Vibrio cholera outbreaks, in Ebola virus, and even TB. Another innovation is bioinformatic approach. So this can be used in conjunction with the NGS to ensure that you detect the genes, AMR genes, and the database can be used for prediction. The field of bioinformatics encompasses a range of computer methodologies, such as sequencing, structural alignments, as well as analysis of extensive biological database. So what this basically does is that as more data is being made available in molecular biology and transcriptomics and proteomics, then you can utilize bioinformatics tools to comprehend this information as we are gathering big data. Bioinformatics allows you to interrogate this big data and then make sense of it by drawing out trends and using that for predictions. So now also we have more recently the AI-assisted pattern recognition as a key biotechnological innovation for disease detection. So what this does is it is emerging as a transformative biotechnology innovation in disease detection and surveillance. Then the pattern recognition refers to its ability to analyze large volumes of complex data, identify hidden relationships, trends, or anomalies. So we know that AI is now being very, is revolutionizing how we use our data because it has the ability to analyze data even faster than we can do initially. And then it can draw out trends. It can look at how disease signals are moving, predict outbreaks, support real-time public health decisions. So this is where AI-assisted pattern recognition is taking the world to. So for precision diagnostics, we have rapid and sensitive detection mechanism. The biotechnological diagnostics have undergone great innovation with tools like CRISPR. CRISPR-based detection later, and nanoscale biosensors that provide rapid, highly specific pathogen detection. These technologies, they enable us to detect microbial DNA and RNA and resistant genes with greater sensitivity than the conventional method we've been using initially. It reduces delay in diagnosis, guiding timely treatment decisions. So let's go a bit deeper on the CRISPR technology and what it is all about. So the full meaning is clustered, regularly interspaced, short palindromic repeats. And it's a revolutionary gene editing technology. It was initially adapted from the bacteria in itself, its own mechanism to resist viral organisms like phage viruses that attack them. It devised a method to actually attack the viruses and cut their genes, enabling it to protect itself. So this technique was now adopted to use in gene editing. It allows scientists to precisely cut, edit, and modify the DNA in living organisms. So it makes use of a guide RNA and the Cas9 enzyme, which serves as the scissors that cuts and edits the DNA. So this fast and efficient tool allows applications in gene therapy, agricultural improvements, and also in molecular research where it is being used widely. So what are the steps in CRISPR gene editing? First, a single guide RNA, like I said, there's a guide RNA. The duty is to, carry information. It is designed to match the DNA sequence of the bacteria, the antibiotic gene, resistant gene that you're targeting. For instance, you want to use that to modify a resistant bacteria so that it becomes more susceptible. So you target the particular resistant gene you're interested in, and this information will be carried by the guide RNA. So the guide RNA now binds with the Cas9 nuclease, which is the enzyme that is able to cut the DNA. It forms a molecular complex with it, and then it searches for the target DNA. So what happens is that when it penetrates the body and it finds the target bacterial genome, recognizes the complementary resistant gene, then it binds to it. As soon as it binds to it, then the Cas9 enzyme will cut the DNA double strand at the targeted gene, then allowing the natural mechanism to repair the DNA. But in the process of repairing the DNA, there is mutation. There will be either deletion or an addition. So making the bacteria more susceptible to antibiotics because it, in the process, loses its ability to be resistant. So this is a way of, artificially trying to edit the DNA of a bacteria for it to now be more susceptible, trying to get rid of the ability to be resistant to antibiotics or drug of choice. So now, and the use of CRISPR has also gone, there are several innovative approaches that it has been used. For instance, in this study, they used it to combat resistance by using it to target biofilms that drive infections. So this advanced gene editing technology has been reshaping approaches to AMR by enabling precise targeting of resistant determinants and even the virulence factors within pathogenic organisms. So it can also be combined with nanoparticles, which is a delivery system that allows the CRISPR components to actually reach the target site. It takes it to the target site and releases it to the cytoplasm. So allowing the CRISPR to now target the gene, bind to it, and cut off the complementary area of the DNA, allowing it to mutate. So this technology is now being also used widely and targeted for other uses. So other emerging technologies include the nanopore sequencing, digital PCR, and machine learning and predictive analysis are also some of the biotechnological tools that are emerging. So now I'll talk a bit more about the nanobiotechnology-enabled therapies. So nanotechnology provides a versatile platform for targeted drug delivery. It allows the drug to be targeted and also in antibacterial action. So that's the area where it's helping us to fight against antibiotic resistance because it allows the drug to get to its target and we have more specific response that is more desirable. So it makes use of also the nanocarriers. There are different nanocarriers that it can use. It can use the liposomes, it can use the polymeric nanopores and the metallic ones. We'll talk a bit about them. But this is a schematic illustration of the different applications of nanotechnology. It can be used in tissue engineering, drug delivery, gene delivery, drug screening, targeted therapy, monitoring biomarkers, and so on and so forth. So you can see that it has a wide range of application in public health. So what are the therapeutic advantages of this nanotechnology? So it improves drug bioavailability and stability because it allows these antibiotics or antimicrobials to reach their infection site effectively. Then it also allows for direct disruption of microbial membranes. So it kills resistant pathogens by mechanisms that are more efficient, unlike the traditional antibiotic that we use. So we also have biofilm penetration. So biofilm poses a lot of challenge in treatment of infectious diseases. But now, with this technology, you can target these biofilms. Nanoparticles allows you to penetrate the biofilms and then release the drug directly into the biofilms, making it more effective. So these nanoparticles may function alone or as antimicrobial agent or as synergistic booster. So in itself, it can function alone or when it is combined with CRISPR components or drugs, it now synergizes and then allows it, takes it as a delivery tool to the target site. So now we're talking about innovative nanotech therapy strategies. So we have these different nanocarriers, but I'll talk about them more in the next slide. So we have the metallic nanoparticles. These ones are either made from gold, silver, iron oxide. They are used extensively in cancer therapy, phototherapy, photothermal therapy, can use the gold nanoparticle. So it functions in the same manner, allows them, it has organic linkers and active molecules attached to their surfaces, and then it delivers them directly to the area where it's needed. And it has a large application in diagnostic imaging for contrast agents using iron oxide. And then it also has application in antimicrobial treatments where we use silver nanoparticles. So, and this is a nano carrier that can also be used. So another one is the polymeric nanoparticles. So these polymeric nanoparticles, we have some of them. Example is the polyethylene glycol and polylactic glycolic acid. So these ones, they have key innovations in health, and they allow controlled drug release system. So because of its ability to release drug at intervals, so you can use it for targeted therapy and a controlled drug release system. So we have the gene and RNA delivery, and then it also has application in vaccines and targeted cancer therapy. So now, for a case study of the application of nanotechnology in vaccine development, a very common example is in COVID-19 during the pandemic. We had the lipid nanoparticle that was widely used during the COVID-19. It played a central role in the development and delivery of next generation vaccines, as seen with the lipid nanoparticle platform. It enabled rapid, effective mRNA vaccine during the COVID-19. So vaccine delivery was very rapid and it was effective because we were able to use the messenger RNA vaccines and using the nanotechnology to deliver this vaccine. So lipid nanoparticles, which is another nano carrier platform, they are advanced delivery vehicles that protect delicate messenger. It has a very high advantage because it's biocompatible. So there is a very low or minimal risk of side effects or body reacting to it. So, and its ability to penetrate different organs and then delivers the vaccine to where it is needed. So this delivery system, it happens by encapsulating the fragile, the mRNA is very fragile, so it encapsulates it, protecting it from degradation and facilitating its entry into cells. As can be seen in the diagram, you can see how it is encapsulating the messenger RNA inside and protecting it. It doesn't, it avoids degradation until it gets to the site where it needs to release it. Then the ionizable lipids would dissolve and then release it into the cytoplasm. So this delivery process, it works through the following steps. First, when the vaccine is injected into the muscle tissue, it binds to the negatively charged cell membrane and enters through endocytosis. So when that has happened, the ionizable lipids, like I explained earlier, will dissolve and release the messenger RNA into the endosome, into the cytoplasm. Then there is protein translation, which the body recognizes this and produces viral spike proteins. These viral spike proteins will imitate the virus itself. So because of that, it triggers the body immune system to start producing antibodies to fight these viruses. So this was used largely to develop vaccines for COVID. So the key characteristics for this lipid nanoparticles.
Hello.
It prevents the
Hello, sorry to interrupt you, ma'am. Ma'am, we are in sort of time, so can you please conclude?
Sorry, I didn't hear you. You said what?
Hello. Ma'am, we are in sort of time. Can you please conclude?
Okay.
Yes, ma'am, please.
So the key characteristics is that it allows protection of the messenger RNA and is very efficient, and it's also versatile. And it's also now being investigated for other bacterial pathogens that are difficult to vaccinate against. So the lessons learned from this whole case scenario was that speed plus precision will always lead to effective epidemic response. So in this table, we try to compare the different nano carriers. And then integrative One Health and future perspective, addressing AMR and infectious threats requires a One Health approach, especially recognizing that the environment, humans and animals, and the ecosystem are interrelated. So these nanotechnology and biotechnology interventions are being explored, not only for human, but also for agriculture, aquaculture, and environmental monitoring. So what are some of the challenges and barriers? We have the toxicity and safety concerns. Because of its ability to penetrate membranes, it can enter and accumulate in different organs. So we do not know what a long-time effect of these nanoparticles accumulation can lead to. So also disposal in the environment can also contaminate environment and the plants and animals. And we really do not know for sure how this can affect the environment in the long run. So because of this, there is need for comprehensive risk assessment, standardized toxicity testing, and safe by design approaches in nanomaterial development is very important. So there are also some regulatory risk, but because of all this risk, WHO and some other drug administration, European medicine agencies are keeping a close eye on this use of nanoparticles to just be sure that it is still used in an ethical manner. For instance, informed consents of patients that participate in such drug trial or clinical studies should be ensured. And then to also avoid potential misuse of this, especially now people using it for non-medical approach, non-medical enhancements. So there are regulations and ethics concerning all this. And then there is need for transparency and public engagement in the use of these innovative approaches. Some other challenges are equity and access to ensure that it is available to everyone, because it can be very expensive and may not be easily available. So in conclusion, these innovative biotech and nano-enabled interventions are redefining the strategies against infectious disease and antimicrobial resistance. So by leveraging their precise diagnostics, gene editing, smart drug delivery, advanced material science, the field is charting a course towards more effective, targeted, and sustainable solutions. So the continued interdisciplinary research is very key to ensure robust regulatory guidance, evidence-based information, and global collaboration, which will ensure which is essential to translate these technologies from the laboratory into the real world use. So these are my references. Thank you for listening.
Thank you, Dr. Chukwu, for your insightful presentation. If anybody in the audience have any question, then they can ask. There are no questions. Thank you, madam. So moving further, it is now my pleasure to introduce our next speaker, Dr. Sunil Mishra. Dr. Mishra is currently serving as Assistant Professor in the Laboratory of Immunology at the College of Life Science, Gachon University, South Korea. He completed his BSc in Microbiology and MSc in Medical Microbiology from Tribhuvan University, Nepal, and later earned his PhD in Pharmacy with specialization in Immunology from Ingham University, South Korea. Dr. Mishra has received several prestigious recognition, including the Gold Award of the Duke of Edinburgh International Award, and has served as an outstanding award leader for the same program. His research primarily focuses on cancer immunology, vaccine development, autoimmune disease, and immunotherapy, with particular emphasis on understanding the molecular and metabolic mechanism regulating immune response. His recent work investigates metabolic checkpoints that regulate T cells, exhaustion, and immune response in cancer, contributing significantly to the field of cancer immunotherapy. May I now request Dr. Mishra for his presentation. Please, sir.
Thank you so much, ma'am, for your kind introduction. And Firstly, I would like to thank Professor Dr. Binay for providing me a wonderful opportunity and the entire organizing team for to deliver my talk in today's conference. So I'm very much grateful. And so, as we are running out of time, I will directly start the presentation. So, you can see, my title is Methionine Consumption by Cancer Cells Drives a Progressive Upregulation of PD-1 Expression in CD4 T cells. So, let's talk about. So when we talk about non-communicable disease, cancer has been the second leading cause of death across the globe. As we can see, first being the cardiovascular disease. And cancer, I think it is very much known to all of us. So I'm not going to introduce cancer detailedly here. So cancer is none than other cells of our own body which are growing uncontrollably. So when the growth of certain cells in our body is uncontrollable, that leads to cancer. And when we come to the treatments that are available at the present moment, we have surgery, radiation therapy, chemotherapy, and among all the various treatments that are available, the most catchy one and the one that everyone is looking for is immunotherapy, because compared when we compare immunotherapy with all other treatments available, it has less side effects than other target therapies. So nowadays immunotherapy is getting more popularity in case of cancer as well as no other disease. So what happens in the tumor microenvironment? So basically in the tumor microenvironment, when the glucose is uptaken more by the tumor cells and it is converted into the lactate, that leads to the acidic environment within the cell. And because of the huge amount of nutrient uptake by the tumor cells, the nutrient availability for the T cells that are responsible to fight against tumor cells is very poor. So that leads to the upregulation of PD-1, which is inhibitory molecule that is expressed on the T cells when there is adverse environment. So due to the increased upregulation of PD-1 in the T cells, the effector function, that is interferon gamma secretion, TNF-alpha and IL-2 secretion by the T cells, is decreased. That leads to the improper functioning of the T cells. So from various research, it had been previously made clear that various amino acids as well as supplements that can enhance the T cell function, as well as some of the molecules can, for example, the byproduct of tryptophan metabolism, canurine, it can inhibit the T cell proliferation. At the same time, methionine is very much essential for T cell growth and cytokine production. So our lab targeted to study the effect of essential amino acids, which was not so much studied. So we started work with in this area. So basically what we started is we started to check the PD-1 expression in case of tumor free mice and tumor bearing mice. So when we check the PD-1 expression in CD4 and CD8 T cells, we can see clearly, compared to the tumor free mice, the tumor bearing mice had more PD-1 expression in both CD4 and CD8 T cells. And we try to replicate the tumor environment in vitro conditions. So we produced tumor supernatant by using B16F10 melanoma cell line. And when we check the PD-1 expression in complete media and the tumor supernatant conditioned media, we can clearly see that the PD-1 expression was upregulated, similar to that of tumor bearing mice in case of both CD4 and CD8 T cells. So we concluded that when there is nutrient deprived condition in the T cell, then the PD-1 regulation, that is co-inhibitory molecule, is greatly enhanced. And we also verified it with dialyzed media, that is the dilation of dialysis of the FBS, and we try to replicate the reduced nutrient condition. So we concluded that when the nutrient condition is deprived, then T cells go in exhaustion. Further, we try to check the effect of other molecules such as glucose, triglyceride and amino acids. So we confirmed that when the dialyzed media is treated with amino acids, then the PD-1 expression is down regulated but it was comparable in case of glucose and Tg in case of CD4 and CD8 T cells. But we directly can see that there is only effect of essential amino acids, not glucose and triglyceride. So we continued our focus on amino acids. So as there are basically 9 essential amino acids, so we treated complete media and dialyzed media with all essential 9 essential amino acids individually, and we can find that only methionine can reduce the PD1 upregulation, which was increased in dialyzed media condition. So that was also verified with tumor soup condition in human conditions. So human blood also, when PBMCs are collected and T cell was isolated, then we can see that in case of tumor conditioned media, that PD1 is very high. But when methionine is treated in case of human being also, then the PD1 regulation upregulation was reduced. So we can conclude that essential amino acid regulates PD1 in CD4 T cells. So further, to continue our experiment, we had one supporting data from Nature. They already reported that CD8 T cells require sufficient amount of methionine to kill the tumor cells. So we checked the expression of SLC43A2, which is the transporter for methionine to tumor cells. So when SLC43A2 was knocked out from the cell line, we can see clearly the PD1 was downregulated in tumor. So the in vivo data, we can clearly see that when SLC43A2 is present, the tumor mass and tumor volume was greatly high. But when the SLC43A2 is knocked out from the tumor cell line, the PD1 expression, tumor volume was less, because the cancer cells cannot get sufficient amount of methionine. So from this data, we concluded that cancer cells outcompete T cells for methionine and increase PD1 expression in T cells. So further on, we carried our experiment with the treatment of methionine. So we treated tumor-bearing mice with PBS and methionine 40 mg per kg intratumorally. So we directly treated in the tumor site. So upon examination, we can see clearly the methionine treatment reduces the tumor volume as well as tumor mass, and the percentage of CD4 and CD8 T cells was greatly enhanced by the treatment of methionine. So it shows that methionine does carry anti-tumor immunity for T cells. Then we also try to check whether only CD8 is capable of killing the tumor cells, or there is some role of CD4 also. So there were two cells treated with CD4 cells treated with PBS and treated with methionine. So PBS cells expressing high PD1 and methionine treated CD4 T cells expressing low PD1. So upon the analysis, we can see clearly the lysis was increased when CD4 T cells were treated with CD8 T cells in presence of methionine. That support our data that CD4 positive cells treated with methionine can support or enhance the activity of killing activity of CD8 T cells. So further, we performed metabolomics analysis to see what happens when the T cells uptake the methionine. So from this data, we clearly see that the methionine as well as its metabolites SAM and SAH were greatly downregulated. That is, their expression was fold change was very less in case of tumor soup compared to the complete media, which suggests that when there is less methionine in the tumor microenvironment, the metabolites are not available for the T cell. That leads to the improper functioning of the T cell. So which was also further verified by the in vitro test. The molecular we can see clearly here. And we also check the PD1 expression in presence of methionine and other its metabolites. So which was clearly visible that methionine and SAM in presence of DM, the PD1 expression was greatly reduced when treated with methionine and SAM, which suggests that the methionine, when uptaken by the T cells, it converts into SAM, and then it regulates the effector function of T cells. So further, we try to check what happens within the cell. What is the next step? So we treated our cells with a .1L inhibitor, and .1L is the sole methyl transfer enzyme which converts H3K70 to K79 to H3K79Me2. That is dimethylation of methionine is carried out by the .1L. So in presence of .1L inhibitor, we can see clear dose dependently the H3K79Me2 dimethylation was reduced, which suggests that there is a connection between methionine and the H3K79. So further, we also checked what molecules or metabolites are increased and decreased in case of the tumor soup and complete media. So clearly we can see in complete media there is less glucose available, but in tissue there is abundant amount of glucose, and lactic acid was greatly increased in case of decreased in case of tumor supernatant, which suggests that uh, in T cell there is very less amount of in tumor supernatant condition there is very less nutrient available for T cells. That is why the effector function of T cells is lost. So when we can also see that from metabolomics the analysis that several molecules like PRKA1, which is the gene responsible for AMPK expression, is down regulated. So we try to check AMPK expression in tumor free and tumor bearing condition, in tumor supernatant and dialyzed media condition. So we can see clearly that when the nutrient is deprived condition, then the AMPK expression is very less, and when the dialyzed media is treated with methionine, then the MPK is expression is reversed. So it supports that nutrient deprived condition leads to loss of AMPK expression in the T cell. So similarly, we also again perform the tumor experiment with wild type and AMPK conditional knockout mice. So in this case, the AMPK is knocked out from CD4 and CD8 T cells. So we can observe that in case of knockout mice, there is increased tumor volume and tumor mass, and the PD-1 expression was greatly high. And when this knockout mice was treated with methionine, then there was no comparable difference, which suggests that methionine exhibits anti-tumor immunity by the AMPK. So it suggests that AMPK is required for by the T cell to control the tumor by its upregulation. So further, we also try to check the effect of ICAR, which is AMPK regulator. So we treated mice with methionine as well as ICAR. So we can see clearly that the co-treatment of methionine and ICAR greatly reduces the tumor volume and tumor mass and the PD-1 expression in CD4 T cells. And we performed similar experiment with RAG1 knockout mice. And in RAG1 knockout mice, we can see clearly that there was no tumor mass and tumor volume difference. So RAG1 knockout mice is that there is no T cells. So this experiment supports that methionine and ICAR exhibit immune cell dependent anti-tumor immunity. So further, we perform the RNA sequencing of AMPK knockout and wild type CD4 T cells. So we can clearly see that the response to genes related to stress, genes related to immune system processing, cell activation, where some genes were down regulated, upregulated, as well as the genes related for cell activation, they were down regulated. So as we can see, the topmost gene which was down regulated which was upregulated was a response to stress. So we thought that there might be the cells might go in stress condition when there is deprived nutrient condition, and it was also verified with the GSEA plot. So we try to check in the downstream of stress phenomenon in T cell. So we can see clearly that XBP1 is a one molecule which was greatly upregulated in case of AMPK knockout. So it gives a clue that XBP1 is upregulated in AMPK knockout CD4 T cells. So further, we performed experiment with XBP1 expression spliced, because there are two forms of XBP1, spliced and unspliced, and XBP1 spliced form is responsible for the stress, to cause stress in the T cells. So we clearly see that in case of wild type mouse, there is, when CD4 T cells are stimulated, only stimulated condition, there is no PD1 expression. But when there is XBP1 is in M knockout, then there is more PD1 expression. And we also use TUDCA, ER stress inhibitor. So when TUDCA is treated, then the PD1 is down regulated. And we also silence the IRE1, which is responsible protein for expression of stress in molecule in T cells. And we checked that XBP1 was expression was downregulated when methionine was treated in tumor supernatant. So which suggests that AMPK regulates PD1 expression by XBP1S. So finally, from this data, we concluded that methionine dependent nutritional control of CD4 T cells exhaustion in the tumor microenvironment, and the consumption of extracellular methionine by cancer cells in T cell exhaustion was upregulated by PD1 expression on T cells, CD4 T cells. And then methionine competition increased SLC43A2 expression in various cells can allow them to uptake more methionine in the tumor microenvironment. And this was also verified with correlation analysis between SLC43A2 expression and survival rate in cancer patients. So it is human genome atlas data. So when SLC43A2 expression is high in the tumor cells, the survival of the tumor patients is very less, and reduced H3K79 in tumor infiltrated CD4 T cells, reduced AMPK expression, and consequently increased PD1 expression. And the down regulation of AMPK under methionine consumption by cancer cells increases ER stress. And our data demonstrate an epigenetic link between the methionine metabolism and AMPK, which can down regulate the expression to enhance anti-tumoral immunity. So coming to the last. So what happens, we try to replicate in the graphical abstract. So when there is low methionine condition in the tumor microenvironment, that methionine is not available for the T cells, then there is lack of SAM, S-adenosine methionine, and that leads to the histone methylation. And because of the less methionine, the T cells express the MPK expression is very less. The cells express PD1 and go to stress condition. Because of that, the cells become in exhausted condition by expressing PD1 more. Interferon gamma and granzyme secretion is very less, and their anti-cancer effects are very poor. But when we supply more methionine that is targeted for T cells, then in case of abundant methionine availability to the T cells, there is less PD1 expression, and then because of the AMPK expression, the AR stress is downregulated, and the T cells can perform anti-tumor immunity by more secretion of interferon gamma and granzyme B. And this data we were successful to publish in Nature Communications in 2023. So lastly, I would like to thank my PhD supervisor, Professor Jae-Hoon Sang, our collaborator, and everyone who directly, indirectly supported us to publish this paper. And lastly, I would like to sincerely thank Dr. Binay Dwivedi and the entire organizing team from MIT University for providing me this wonderful opportunity. Thank you so much.
Thank you, Dr. Mishra, for that highly informative presentation, for sharing your important research insights. If anybody from the audience have any question, then they ask from the sir. There are no questions. Thank you, sir. We now move to our next invited speaker, Dr. Feroz Khan. Dr. Feroz Khan is a senior principal scientist and professor at the CSIR Central Institute of Medicinal and Aromatic Plants, Lucknow, one of the India's premium research institute under the Council of Scientific and Industrial Research. Dr. Khan completed his PhD in bioinformatics from Uttar Pradesh Technical University and further pursued postdoctoral research at the Sparks School of Pharmacy and Pharmaceutical Science, University of California, San Diego, USA. His research expertise spans computational molecular biology, bioinformatics, machine learning in drug discovery, transcriptomics, and chemoinformatics. Dr. Khan has an outstanding scientific record with more than 120 research publications, an H-index of over 35, and thousands of citations, reflecting the strong impact of his research contribution. He has also been involved in more than 30 research projects funded by national agencies such as DST, ICMR, CSIR, and DHR, and has delivered over 100 invited lectures in national and international conferences. He is actively associated with several professional bodies, including the Society of Biological Chemistry, India, and serves as an editorial and advisory board of scientific journals. May I now invite Dr. Khan to kindly deliver his presentation. Please, sir. Sir, kindly proceed for a presentation. Please, sir.
Hello,
Yes, sir, you are audible and your presentation is also visible here.
can I start?
Yes, sir, please.
Okay. Dear participant and the organizing committee, good afternoon, all of you. And thank you for giving me the opportunity for delivering a talk on Introduction to Quantitative Structure Activity Relationship Modeling and its application in lead identification and lead optimization process of drug discovery programs. Myself, Dr. Firoz Khan from CSR CMAP. I'm working as a senior principal scientist here. And my research domain basically on structure activity relationship and bioinformatics. And structure activity relationship is a part of like a drug discovery program where we try to find out the potential lead based on phytomolecules and their derivatives. So starting with this topic, I just summarize some of the basic points of the QSR modeling and its application parts. As you see in this slide, I hope you are able to see this slide. This is a graphical representation of the process of the QSR model development, which is a machine learning method basically used for high throughput data screening of especially the phytomolecules and the derivatives. In the process of like developing a model, you should need a training data set, and you have to train the model in such a way that you get some significant statistical parameters so that you can accept it. After developing a model using either the regression approach or the classification approach or the optimization approach, you just try to screen out the query set molecules and predict the activity based on the models. So it gives the idea of like optimum value of the activity of unknown or untested molecules. So before on clinical trials or preclinical trials or in vitro, in vivo studies, we normally virtually screen a lot of molecules before going for wet lab validation. This way we can save the wet lab economy and also streamline some of the potential molecules rather than testing junk molecules. So this is a very time saving and economical approach. And nowadays everybody means using this approach, and worldwide, even in India, different labs are using it. If you see the definition of the QSR is it is a process by which biological activity is correlated with physicochemical properties of the compounds, and these compounds are part of the training data set. So every machine learning model, supervised especially need a training data set to learn the model. So this way we can train the model in such a way that can utilize the prediction power and predict the values of untested molecules. Its application part is basically in drug discovery part basically, and also the toxicity prediction or the property predictions. You can develop QSR, QSPR, QSTR models and utilize as per your need. It uses both mathematical expression as well as machine learning methods. If the data set size is more than 200, 300 on thousands, you can utilize machine learning methods. But if the data set or sample size is very less, especially in natural compounds, you can utilize a statistical approach like regression, correlation and collinearity for developing a model. This approach is first time utilized by Corbin Herman Hensch, and it is considered as like father of computer assisted molecule design. First time he developed the QSR model, and this model equation is based on line equation y is equal to mx plus c. As you see in the presentation, this equation is called as Hensch equation, and it is developed in 1969 first time, where he successfully correlated biological activity with the log p parameter, which is a representation of bilipid membrane or hydrophobicity permeability. In machine learning method, if you see earlier, we use a programming skill, but nowadays already the program is developed. You have to just utilize these method in such a way to just give input data and you get the output data. So the comparison of traditional method as well as machine learning method is there. You see in the, you see in the traditional approach, we provide the data and the program as an input, and traditionally programming method utilize these two parameters and give the output at the end. But nowadays machine learning utilize in such a way data that we provide only input data and output at the same time as a sample size, as a sample, output as a sample, and input data is provided based on training data set. Both data is provided without any program, and machine learning method learn it and give the output. And this is called the model, or what we call as the program or logic. And these outcome is part of the research nowadays. As you know, nowadays it is the artificial intelligence boom is there, and as you see in this diagram, artificial intelligence covers two major components: machine learning and deep learning. So deep learning is a subset of machine learning, and machine learning is a subset of artificial intelligence, and each set covering a lot of methods. Deep learning utilizes the artificial intelligence method, ANN with a lot of hidden layers. And similarly, machine learning method utilizes both supervised and unsupervised method. And also within the supervised method, there are so many other methods. I will just highlight in a next slide. So you see, a machine learning data set required low training data set, while deep learning required high training data set. So accordingly, performance and training data set requirement base, there is a graph representing, which indicates that machine learning method training data set size is low, but performance is not so much high as compared to deep learning. Similarly, as I mentioned, machine learning methods cover so many methods basically, but in research areas, only 2-3 methods are basically used. For example, supervised and unsupervised method. Reinforcements we do not use at now because we don't have such training data set available right now, but in future it may be possible. Similarly, within supervised machine learning method, there are two methods: classification method and regression. So normally we utilize classification. A lot of research lab universities utilize these methods. Within classification method, we use decision tree, Bayesian learning, random forest, logistic regression, KNN, rule induction, and so on. SVM, ANN, you heard about these methods. So each method have pros and cons. Based on your training data set size, you can utilize each method and compare the performance. Based on performance, you can select the method. Similarly, regression methods are again no so many types like linear, simple linear, multiple linear, non-linear. So based on your data set, you can utilize each method. Similarly, unsupervised method, you see, clustering is very commonly used method. Clustering, within clustering, there are k-mean clustering, hierarchical clustering, dimensional reduction method, which is also called as PCA. So these methods are very commonly used in bioinformatics and other biotechnology methods. In QSR, basically, we utilize the method, which is, I just listed out some of the points, like you have to go through with data collection and then data cleaning or pre-processing, then model development and training and testing and tuning. Tuning is optimization. And lastly, you can utilize this method for prediction or classification. There are so many other components of the machine learning where machine learning algorithm comprises three components, basically representation of the data set and evaluation of the model and optimization of the model. So within representation, you see there are so many methods I've listed here. So decision tree is very commonly used method in representation of data, set of rule or logic programs you can develop. Instances you can consider Markov or Bayes method, you can consider neural network, support vector machine. And similarly, for evaluation of the model, you can utilize so many other methods like likelihood is very common method. You can utilize that. And this way you can select whether your model is working fine or not. Similarly, optimization methods are there, greedy search, gradient descent, and linear programming. So based on your purpose of the model, you can utilize these evaluation and optimization method and go for further analysis. This is a graphical representation where we utilize the training data set. And the first half you see, we utilize the training data set, 80% of the training data set utilized as a training data set and develop the model based on independent and dependent variables, parameters, and develop the model. And after that, we test the model using 20% of the data, training data set, which is also called as testing data. And we see the performance. And we compare the performance in terms of error, which is also called predicted results minus actual data results. So we get the error and we try to minimize the error. And when we get lowest error, then we select the model. So this is a very common approach in developing any QSR or QSPR or QSTR model. There are some steps in the QSR process. As you see, you need to have structure, chemical structure in linear, 1D, 2D, and 3D form, and try to calculate the properties, chemical properties. These chemical properties called as constitutional properties or descriptors, electronic descriptors, properties, geometrical, hydrophobic, lipophilicity, quantum chemical, topological, and so on. So there are so many open source softwares available you can utilize, like Paddle you can utilize for prediction of these chemical properties. It is freely available. There are commercial softwares also. You can utilize AlphaDesk, for example. You can utilize this for chemical descriptor calculation. So one compound can you can get 1,000, 2,000, or 3,000 chemical properties. After getting the descriptor calculation, try to normalize it, standardize it, and clean the data by removing the outliers and correlated descriptor, remove it. After that, you just try to make the relationship using the multivariate analysis. Any method like multiple linear regression method or self-organizing map, PC analysis, partial least square method, neural network, or support vector machine. And then based on statistical parameters like regression coefficient, correlation coefficient, and cross-validation correlation coefficient, and mean standard error, and root mean square error, and so on. After reducing the error, we can check the performance in terms of predicted activity versus experimental activity plot. And after getting the linear arrangement of the data points, we can able to select the model and set the applicability domain filter. And this way we can predict the activity of untested molecule before wet lab analysis. This is also called as virtual screening method for lead identification or drug discovery. As you know, molecular descriptors is a chemical properties of the compounds, organic compounds or active compounds. We try to calculate the properties of each compounds and then try to correlate with the activity. So these are numerical values as mentioned here and that characterize the properties of the molecules. We try to find out the those properties which have high correlation with the biological activity. You know, basically there are so many properties which having similarity with each others. So we can remove these correlated properties of the chemical compounds by correlation matrix, or you can utilize multicollinearity analysis, or removing the same class of the descriptors. There are so many approaches. Even PCA you can utilize this. I just listed out 5 major classes of the chemical descriptors. You can select 1 or 2 properties from each class and then try to develop the model. Then I hope you will get the correlation well and develop a good correlation model. So these 5 classes are basically constitutional descriptors as mentioned here: electrostatic descriptors, topological descriptors, theoretical descriptors, quantum chemical descriptors. And based on softwares available right now, I just mentioned some of the software as you see on the top first row and different columns: Moderate, Paddle descriptors, Bluedesk, ChemoPy, Pi, DPIs, RCPI, SynFeed, Dragon. You can utilize any software for prediction of the chemical properties. Some are freely available, some are commercially available. And try to make the correlation matrix as mentioned here. Each column represent properties and you try to remove those properties which have high correlation with each others. But property should have correlation with the biological activity, but not the property correlation with the same property. So property, we just based on this correlation matrix, we remove the same or homologous properties and try to find out the highly correlated properties which have correlation with biological activity. So this way, this is a representation of the properties where how we calculate the linear regression using least square method. And as you see, x is basically independent variable, y is dependent variable, and just try to differentiate it and then try to find out the mean value and differentiate it and then calculate the sum of these values and put on the formula and we get the correlation linear equation, which is also called as y is equal to mx plus c. And plot it on the graph as you see here. And based on that, we can calculate the different coefficient values of the regressions. Similarly, we calculate the R square and regression coefficient value using the same formula as it is mentioned here. You see, and then after getting the lowest distance error between the predicted and experimental, we can go for further iterations and cycles. And after getting a lowest R square value in terms of error, we just select it, which is also called a sum of a squared error. So low is the sum of a square error, high is the correlation coefficient. So this way we can calculate the different properties and correlate the different properties with the biological activity. There are so many advantages of the QSR. As you see, earlier we screened randomly compounds and try to find out which is active, which is not active, also called as a structure activity relationship. Now this term is also known as quantitative structure activity relationship or 2D QSR. And there are other method of QSR. It is also called as 3D QSR, where 3D structure is required. But most of the time we don't have 3D structures like having a target with their bound ligand or known drug or phytomolecules. So we prefer 2D QSR and then go for 3D QSR. So both the methods are similar in nature, but 3D QSR required 3D PDB database ID and crystographic data information. As you see, advantages indicates the understanding of the effect of the structure on activity. And also the QSR is useful to make the prediction leading to synthesis of novel analogs. And also QSR helps to understand interaction between functional groups in the molecules of greatest activity with those of their targets. So you can also find the targets also at the same time and validate through molecular docking and MD simulation studies. At least 100 nanosecond simulation is required. Some of the commercial example where drug is discovered based on using the classical QSR. Classical QSR means 2D QSR approach, and these are in market available. These drugs are available in market. These are I listed some of the drug: norfloxacin, bromobutide, and glomarizine, and metconazole, fluvophene, and there are so many other drugs. I just listed some of the reported drugs only. You can explore all these drugs in Google's, and you can find different literature also which listed these drugs. After so many means application part, as I mentioned earlier, there are some disadvantages of the QSR. New user or the student or those expert who have no idea about the QSR model development and try to start a career in this field normally done a lot of error in the initial phases, which I observed during my career, and I found there are minor mistakes create a huge blunder in the analysis. So some of the disadvantage I listed here and try to develop the model with the help of taking expert help. Without any expert help, you cannot develop the 2D QSR model because it gives some biasness. So there are some possibilities, some errors are there, like false correlation may be possible during the analysis, and very short training data set sometime collected, and you develop the model. This will give the error at the end. Similarly, the selection of the chemical descriptors is again a major problem. How to select the chemical descriptors? Which chemical descriptors is good enough? We don't know actually. Similarly, the selection of the chemical compounds or the training data set is a major challenge, and normally mistake done in this area. And lastly, the statistical parameters normally used is not good enough to indicate the error. So parallel, you should utilize 2-3 method, and then after developing 4-5 models, and then after comparison you can select the best model. Without comparison you should not go, otherwise you will get the error at the end. You will get no result in vitro lab validation. I just listed some of the example which where we done analysis and published it. It is already in the publication, and this is example of you see QSR model development or galvanic acid derivatives for immunomodulatory and anti-inflammatory activity, where we utilize the LD50 value and convert it in log form, which is also called normalized form, and then predict the activity in LD50 forms. This where we utilize some of the known anti-inflammatory immunomodulatory drugs and their derivatives, and in the gallic and in the query set we utilize the gallic acid and predict the activity. And finally we got this model as you see here with the cross validation of R square regression coefficient of 0.96, and after experimental validation we got exact same activity as reported in predicted model. So this way we can screen a lot of galvanic acid derivatives and screen only potential well. And now these galvanic acids utilized in herbal formulation and preparations in CMAP. Similarly, this is an example, another example where we showed regression coefficient and cross validation coefficient of 0.9, 0.89 and 85, where 89 indicates the prediction model performance 89%, and cross validation indicates the performance of model on predicted molecules, and it gives 85%. You see the trainings data set site regression plot is overlap with the actual and predicted same. Similarly, here the actual predicted plot is indicated, overlapping indicates 100% match, and this model is good enough, and it is based on 28 training dataset compounds tested on test 6 molecules with these 4 properties. These chemical 4 properties are highly correlated with the activity, and we finally predicted the activity of 1.941, a log inhibition percentage inhibition ratio, which is experimentally found exact same in lab, wet lab, 1.972. So exactly we screen the good molecules with this method and develop the herbal formulation later on. This is another example where QSR model indicates good enough, but as I mentioned, there are disadvantages. Sometimes we could not see the graph. You see, this is the graph where we don't see any overlap, like red color lines indicating no match with blue color lines. So this is a test data set which shows no overlapping. So there is a high chance that it is a biased model and it shows error. So this way, although the training data set indicates overlapping with each other, actual is indicated by red color line and blue line indicated as predicted. You see predicted and red color lines are almost similar in overlap, but test data set indicates clearly that there is a no overlap. So this way we can reject it. So we rejected is although the regression or the statistical parameter is good enough. R square is 0.85 and Q square is 0.79. Even the plot, linear plot, line equation plot indicates good linear arrangement of the data points of predicted and actual data points. And with using the training data set of 27 and test set size of 7 compounds. So sometimes it is possible that we could not see the loophole. So try to discuss all these parameters with some expert before finalizing the model. This is another example where training data set size is too much high. For example, we have taken the bioassay record of high throughput screening containing 2,291 compound from NCI USA database.
Sorry to interrupt. Sir, we
Yeah.
are in short of time, so can you please conclude?
Okay.
Yes, sir.
So are there participant? I am not able to see the participant. Even in the hall, I am seeing no student is there.
Sir, students are there, some participants are there in the auditorium with us.
Okay.
Yes, sir.
So you see, there are a lot of molecules utilized, and as I mentioned earlier, that if the training data set size is in thousands and 2,000, 3,000, you should utilize machine learning method. So we use virtual method, machine learning method, which is also called a support vector regression (SVR), and develop the QSR model. And these all compounds are anti-cancer active compounds active on breast cancer. And you see, after clustering, we develop 8 clusters indicating centroid molecule on the top, and each clusters have 60 to 50 molecules in their training data set. For each cluster, we develop the QSR model and then screen the molecules. This way, we can predict the activity. And this is also published. Some of the open source and source softwares and free tools are available for QSR study. For example, RDKit, OpenBabel, and Paddle Descriptor and Dragon, Coral, and TalkStree, QSR Co. You can utilize these model and softwares and already existing developed model for virtual screening of the molecules. Similarly, there are commercial and advanced platform. You can utilize like Schrodinger software, MOE, Discovery Studio, CSAR, ICM Pro, and it is available in India also. You can purchase it and utilize for drug discovery programs. Similarly, there are 3D QSR related tools, Codesa, Topomar, Elmont, Holograph, and you can utilize these softwares and for 3D QSR model development. There are so many other open source toolkits available for the beginners, for the industry and a specific task, you can utilize through Schrodinger, MOE, Paddle, RDKit, Python and R packages are there. And there are open source analysis methods are available on Galaxy. You can utilize that platform for a starting career. With this brief presentation, thank you very much. If you have any question, you can ask.
Students and participants, if you have any question, then you can ask to the sir. There are no questions, sir. Thank you so much for your informative talk.
Okay.
With this, we come to an end of session 2 of Bio Nano 2026. We sincerely thank all our distinguished speakers for sharing their valuable research and perspective. We also extend our gratitude to the chairperson of this session, Professor Dr. Vinay Dwivedi, Director AIB, for chairing the session so efficiently. All the students and participants are requested to reach Seminar Hall of Block D for valedictory session. Please proceed. Thank you so much. Students, your attendance will be marked there at Block D, so please be there.